yesterday we were discussing about the OTU picking. Can you remember these things? Three different methods. One is de novo, okay, and another is closed reference.
This like
okay. So and open reference is first closed reference, then go for the de novo with the rest of the reads. Okay. So we talked about these different databases. You can use these databases for taxonomic assignment. And as I told you, there are different statistical analysis approaches are there, and different visualization results you can actually use. So yesterday we have seen the microbiome analysis, okay, where you just have to upload the OTU files, metadata files, if possible, the taxonomic files, and those things. If you do, it will create the different results. So for here you can see KIME output includes a representation of a taxonomic tree in unique format, which can be visualized in application like Fig Tree. This is another freely available software, and a file of biome, which is biological observation matrix file. Okay, so format representing OTU table. So you nowadays you can change this OTU table is kind of old concept in KIME. Now you have to change it to the biome format. Okay. So there are several free softwares available within the KIME itself, which you can actually apply in order to transfer, change one OTU table to biome format. Okay. So this file can be imported. Megan is another like KIME-like software where you can do this analysis. Okay, microbiome analysis. And what are those analysis? First thing is you need to normalize. Okay. So yesterday you have seen the filtering is one of the most important thing. You need to filter first. Okay, number of low reads and other things you need to just throw it out. Then you need to do the normalization. Okay, so normalization is very important, because unless until we do normalization, we cannot compare between different samples. Okay, so we need to have TSS normalization or other type of normalization, which is already available in this softwares. Then once we do the normalization thing and other thing, then we go for different types of this diversity analysis. Okay, this is mainly alpha diversity. That is the diversity. Oh, okay. So alpha diversity, which is the diversity for a particular sample. Okay, like alpha diversity is sample-wise diversity. And you can see different diversity indices are there. We will discuss about all these things like Chow, Sampson, Shannon index, Simpson index, pollinating diversity, etc. Different diversities are there, and each of the diversity indices will give you different interpretation of the data. Okay, and then beta diversity. The beta diversity is actually the diversity between the group of samples. Suppose you have a group 10 samples, group 1 and group 2. Then if you want to check whether between these two groups the diversity is significantly different or not, then you will calculate the beta diversity. Okay, so beta diversity is diversity across the samples, and there are Bray-Curtis indices are there, then Unifrac and other. We will discuss these things in detail. So as well as taxonomic composition, phylogenetic analysis, so all these things you can do by Megan or KIME. Nowadays KIME 2 is there. So recently this R statistical programming language has gained immense popularity and is currently widely used for multivariate statistics. Packages such as Vegan, Phylosec. So Phylosec actually you can create. Yesterday you have seen all these profile plots and other things. Those were mostly created by Phylosec. Okay. So it's another freely available software. You can just upload the package in R and you can do the analysis. Bioconductor is another kind of package providing multiple inbuilt functions. So you just have to call the package, and different inbuilt functions are there. Like you just have to call the library, then mention like alpha diversity, Chow, and then you have to give your data. Okay, and it will do the analysis for you. So this is the figure of a OTU table. Okay, so can you see it's kind of a matrix? In Excel format you can see So here these are the samples. Okay, sample 1, sample 2, sample 3. And these are the OTUs. OTUs means one OTU represents a particular taxonomic trait. Okay. So this is the different organisms. Okay, OTU 1, 2, 3, 4. So here the tree what we were talking about, okay, the Nimiec format tree, the tree will be this taxonomic tree which is actually giving the correlation among those OTUs. Okay, so this taxonomic tree will give you the phylogenetic kind of relation between all the different OTU sequences. Okay, and these OTUs are representing a particular cluster as we discussed yesterday, and we are only considering one sequence representative, okay, from that cluster. Okay, so if the OTU is kind of say only present in 1 or 2, then yesterday you have seen we have to discard all these things. Okay, so very low abundant OTUs we have to discard. And you can see, so if we calculate the diversity within this sample, that will be alpha diversity. Okay, but here you can see we are comparing between the samples, that is called beta diversity. Okay, so it is often assumed that read counts in OTU table are approximately equivalent to observation per species in traditional equality. However, interpreting OTU read counts is actually much more difficult because of biases and errors produced by PCR and sequencing. So you will get different counts here. This is normalized values you can see. Okay, but actually you will get different counts here, like 10, 100, 50. Some counts will be there. Okay, so those counts you can think of, they are representing the species abundance in any sample. But most of the time different errors due to PCR bias error and other errors will be there. So we need to remove all these errors first of all. Okay. So here you can see the count is represented as 0 to 1 is kind of abundance. It's giving the abundance. So you can either normalize it based on suppose sample 1 you have 10, 20, 30, 40 something like these OTUs. Now you can normalize with its total value. You can divide with the sum for each of these values and you can get its ratio. Okay. So this is called the summation normalization. Okay. For a particular sample, you have to normalize by taking its total number of counts. Okay. And then divide that value with each of these values. Okay. Like suppose here 10, 20, 30 something is there and total value will be 100. Then it will be 0.1, it will be 0.2. That will be the normalized value. Okay. So you have to do it in this columnized manner for all the samples. You don't have to do it, the program will do it for you. But you have to understand the thing. You are not working with directly from the abundance. You have to normalize the data throughout the OTU table. That's the important thing. Okay. So now it's another kind of algorithm for UPerse OTU algorithm. Okay. So this is the UPerse method by which we just create these OTUs. You can see here, looking at this picture. Okay. This is done by Edgar. Okay, he's famous for many like CD Heat and other many softwares. If you go to his website, you will get lots of this clustering software. He is quite phenomenal in developing this kind of clustering softwares. He has lots of clustering softwares. Okay, so here you can see. So like this is the OTU sequence in middle of a cluster. That are clustered, you can say cluster centroids. Okay, so these and these are different more than at least 3% level. Okay, so these are the nucleotide sequences. Yes. Now if you align that to nucleotide sequence, you can easily get to know the identity and mismatch between them. Okay, and you can calculate the percentage of identity and mismatch. Yes. So this is a cluster. And the cluster definition is such that look here, the nucleotide diversity between these and these is more than 3%. Okay, and these should be more than 3%. These and these should be more than 3%. But these neighboring OTUs, they have similarity or differences less than 3%. That's why it belongs to this particular OTU. Okay, and you can see here.
In all the cases, it is 3%.
No, so here we are considering the threshold 97%. That's why it is 3%. If you will be considering like 99%, then this will be 1%. Okay. So you can actually think of a 3% radius. Okay, based upon this centroid kind of sequence. Okay, and based upon the 3%, this will create a OTU. This will create a OTU. Okay, this is a chimeric sequence. We will discuss about that thing later on. So this is, we have to discard all these chimeric sequences. And these are some problematic OTUs which actually matches with this one as well as this one. Okay, so these are some problematic OTUs, and it will, program will look its nearby neighbor, and based upon that it will assign to this one or that one. Okay, but it cannot be assigned in both. It will be assigned only in one OTU. Okay, not in the both. So the algorithm constructs a set of OTU representative sequences from NGS amplicon reads. The reads should be pre-processed to overlap pair reads. So that's an important thing. Like in case of, so in case of So in case of Illumina reads, you have paired reads, but in case of nanopore and others, you will not have the paired reads. So there is no problem at all. Okay, but what happened in case of paired reads? Okay, in case of paired reads, you have to merge the reads like this way. So this is R1 read, this is R2 read, and based upon this overlap, because you are sequencing this thing in overlap. Suppose this is the sequence, overall sequence. This R1 read is sequencing this way, and this R2 read is sequencing this way. So now you have to merge these two things based upon this overlap, so that you will get the merged sequence. So you have to work with this merged sequence in order to create these OTUs. But in case of this nanopore sequencing and other, whenever you will get the whole sequence together, then you can directly work with that. In nanopore, even you can get the whole 16x. Okay, the 1.5 kb 16x you can get. Then again the thing is you have to cluster, and other things are similar to this. You have to cluster these things, and you have to assign the taxonomy and other things. So all the things are similar. Only the difference is in case of Illumina there is paired reads which you need to merge. For others which will give you the whole reads, you don't need to merge. Clear? So here you can see. So here you can see you first have to merge the reads. So reads should be pre-processed to overlap pair reads. Is appropriate. You have to remove the barcodes. Obviously, barcode should not be there. Perform quality filtering. Okay, and global trimming. Okay, if any bad places are there at the base or something like that, you have to trim those things. So post processing is needed to map reads to OTU and construct a OTU table. So what are the clustering criteria? The goal of the UPers is to identify a set of OTU representative sequence, okay, a subset of the input sequence satisfying the following criteria. What will be those criteria? If you select 97% sequence identity threshold, then all pairs of OTU sequence should have less than 97% pairwise sequence identity. Okay? And OTU sequence should be most abundant within a 97% neighborhood. So you are making a 3% radius. Okay, 97% similarity means 3% radius of differences. Yes? Then chimeric sequences should be discarded. All non-chimeric input sequences should match at least one OTU with greater than equals to 97% identity. That means here your motto is to build these type of clusters, and you need to expect from, you have to exclude these chimeric sequences, and all other sequences should be included to particular clusters. Okay, this is a very fast program, and it will again do the clustering in a very faster way. So there is another program is called U-noise method. It's a very nowadays people are using these things. This U-noise algorithm, this performs look error correction or denoising. Okay, so it will look for the errors present in your data. Okay, how to identify the errors present in your data? Like I am showing one particular slide is there. That is easier to understand here. Okay, suppose this is your original sample. Okay, species 1, species 2, 3, 4, 5, 6. So all total 6 species were there. Okay, clear? Now what you did? You did the PCR amplification. After PCR amplification, what will happen? These are the PCR errors. One change might happen, but you see the mostly big thing is there, but small changes will happen. These are chimeras. You can see both the colors are there. Okay, these are the chimeric sequences. So this is omitted. Look, it was present in very small amount, and whatever depth we actually consider, that is not enough to sequence this thing. Okay, so this is being discarded. And this is say primer bias. Suppose the primer is pretty much attaching to these things, but that primer may not attach to this 16S RNA. Okay, something is happened, some changes are there in its 16S RNA. So this is called primer bias. So now after sequencing, what you will get? You will get, look for this thing, you will get a big cluster, then the smaller ones. So from this you will get these sequences and these sequences. Okay. Due to sequencing errors, you will get many small fragments. Okay. Now what will happen? These are called tag jumpers. This is also happened due to PCR. Whenever we add tags, so tags from two different samples may jump together and create these tag jumpers. You also need to remove these things. So whenever you do, after taking this thing, you do the quality filtering, trimming, chimera removal. Then if you do clustering, say 99% or 97%, what will happen? You will get 1, 2, 3 clusters. But we had 6 species. You are getting 3 clusters. Okay, so centroids, this is one centroid, this is one centroid, this is one centroid. Okay, so this is a big problem. But in denoising, what you will get? You will get 1, 2, 3, 4, 5. 5 haplotypes if you use the denoising. So at least, obviously you are considering these tag jumpers here, but at least you are considering all other species present here. So this is the advantage of using denoising method instead of using clustering method. Okay. Because clustering method you are considering 97-99% similarity, it will create a big kind of circle, and it will take very close by species within, like this species 3 is engulfed by the species 4. That's the problem in 97% or 93% clustering. So people use the denoising method, and this is actually the method for data 2 also. Okay, we discussed that thing later on. So here, if you think the U-noise algorithm, it performs error correction. What is error correction? This is called the denoising. On amplicon reads, each of that. Correct biological sequences are recovered from it. So here the idea is you have to discard the PCR biases, sequencing errors, or all those things. Those will be marked as the noise. Okay, you need to remove all those noises. Okay. So, distinct sequences down to a single difference or 2 or more difference almost always. Errors are corrected as follows. Leads with sequencing and PCR point error are identified and removed. Chimeras are removed. Now, see here. Okay. So, this is suppose one big sequence. So whenever you got like a chunk of sequences, similar sequences like 20, 30, 50, 100, 1000 numbers of similar sequences, that's a real one. That's a real one. Okay, like this size is bigger, means more numbers of reads are present here for this thing. So this is a real one. Okay, now what you will do, you will create a kind of circle of changes. This is one nucleotide change, this is two nucleotide change, this is three nucleotide change, something like that. Okay, now look here, what the program will mostly do, it will go to this layer and look for again the big chunks. Okay, these two big chunks are there. So it will just put these things. It will omit these smaller chunks as errors because they are present in only one or two numbers and only one nucleotide changes are there. So they will be omitted. But unfortunately, so the program can easily identify this one perfectly. But this is somehow due to PCR error, a large chunk appear that will also be considered here. But all these small chunks will be omitted. But otherwise, if you do the clustering method, what will happen? It will take all these things in a single cluster and it will give a erroneous result. It will consider all these things as a kind of organisms representative of the organisms. So that's the beauty. This is the denoised algorithm. Okay. So like one nucleotide change, two nucleotide change, and very small amount present. Those will be the noises. Those will be discarded from your data. Okay. So this is the thing. It's described here in detail. So like here the left panel shows the neighborhood close to a high abundant unique read sequence like X grouped by the number of sequence differences. These are the differences. Okay. Dots are unique sequences. The size of the dot indicates its abundance. Mostly this is a big, this is a small. So you can see these are unique sequence in the sense that there is one nucleotide change from this here. Okay. So you need to just based on the neighboring method, you need to remove all these things. So then you will get again this OTU. Now there is another thing I already told you, biome format. Okay. OTU, you know that OTU is this one. Okay, it's like a table, tab delimited file. So all these things are separated by tab. Okay, so you can easily open it in Excel. It's .txt, you can just name it. Okay, but here the biological observation matrix or biome format is there. So kind developer OTU tables are represented in a newly developed biome format. This is a kind of Java derived format, JSON derived format rather than the previous tab separated format. The OTU was tab separated format. Okay, so JSON is a language independent data format. It was derived from mostly JavaScript. So several reasons are there because of this change. First, to facilitate the efficient handling and storing of very large OTU table. If the OTU table is very large, look here in OTU table, you will be counting all zeros here. Because most of the OTUs they are present suppose in 20% of the samples, but in others there will be 0, 0, 0, 0, 0. So the OTU table size will be very big. Okay. Now there is a sparse OTU table where the zeros will be omitted. Okay. So those things are converted into this biome format. So efficiently you can handle the OTU table. Okay. And second, to support encapsulation of the core study data in a single file, and third, to facilitate the use of these tables between tools, and I mean you can use in the Kind, MG all different softwares nowadays, they actually consider the biome format files. Okay, not the OTU files anymore. They consider the biome format files. So the thing is, you need to convert the OTU tables in the biome format. Okay. And it is already incorporated in the Kind too. And here is the biome format. You can get the Python code from here also. So they also, these Python codes also can just change the format from OTU to biome, biome to OTU, something like that. They can do it. Okay. So converting between biome and tag delimited table representations, note that you can always use a biome convert command in the biome format package. It's a kind dependency. So you already have it installed to convert biome files to tab delimited text for use of spreadsheet programs. Okay, that you actually can do. Now we will be talking about the chimera. Okay, chimera sequences. As I told you, so can you see these are the some organisms which you never seen. Okay, have you seen these organisms? So this is a mixture of some parrot and some lizard. Okay, so this is from duck and porcupine, something like that. Okay, so think of this kind of it's a mixture. Okay, so chimera is something like that. It's a kind of monster. You have these two biological sequences from where due to PCR you will get this chimeric format. One part from one biological sequence and another part from another biological sequence. And this thing you have to remove from your data. And fortunately there is a chimeric database, huge large database is there. You don't have to do anything by yourself. The program will do everything for you. Okay. So this is the like parent child and parent 2, and this is the chimera is formed, and a chimeric template is created during one round, then amplified by subsequent round to produce chimeric amplicons that will be sequenced later on. Okay. In sequencing, we typically find that only a small fraction of reads is chimeric, perhaps of the order of 1% to 5%. Okay. However, when the reads are clustered into groups of unique sequences in OTUs, then you often find that a much larger fraction is chimeric. So we need to remove these things. Okay, this is a challenging problem in sequencing analysis because chimeras often have low divergence that are very similar to one of their parents. So you need to carefully remove them. Okay, and this is kind of Illumina thing I told you. This is your target. This is only the sequence, and see. This is the reverse forward primer, this is a reverse primer, this is the indices you had, because you have to multiplex that sample. You have to take the DNA from different samples, you have to tag them each sample, and then you have to code those things. Okay, same thing is true for nanocode also. Okay, you have to tag them, you have to index them. Okay, in everywhere, but here only you can do the paired end sequencing. Okay. So I discussed this thing. So the OTU picking, again I am just telling you, this de novo part is there where reads are clustered based on the similarity to one another. It's pretty easy. Okay. And reference based, there are closed reference. Any reads which do not hit a reference sequence are discarded. So in reference, closed reference based, so here you already have a database. Based on the database, you are looking and matching your data. Whichever is matching, you are just keeping them. Whichever is not matching, you are just throwing them. Okay. And open reference, what is it? It is doing like any reads which don't hit a reference sequence, again clustered in de novo way. So it is like a combination of closed reference plus de novo. Okay. And de novo is all reads are clustered. That's the good thing. But the bad thing is you cannot do the parallelization here. It will take huge amount of time. OTUs may be defined by erroneous reads. That's the biggest problem. And for the closed reference, it's good. Built equality filter is already there. So easily you can do parallelization. OTUs are defined by high quality trusted database sequences. These things are good. But reads that do not hit reference data, say some new reads, that will be excluded from your analysis. That's the biggest disadvantage of this closed reference OTU. Okay. And that's why this open reference is good. All reads are clustered and partially parallelizable. So a mix of high quality sequences we define in the OTUs and possible low quality sequences. So these problems should be there. So nothing to worry about. But open reference is the choice you should go for. Okay. So I have already discussed this thing with you. Clustering and denoising. Okay. So this is a UNoise program. So this is the overview of generic amplicon workflow. Now you can see here. So now from sequencing facility, what you will get? You will get the first Q files. Okay. Now the first Q files, the first thing you need to do, demultiplexing those sequences. Demultiplexing means you are pulling all the samples together. Demultiplexing means you are again separating. Okay, that's the demultiplexing. Mostly machine used to do it for you. Okay, then you did it. You started quality filter, quality check and filtering. Then you will get fast affines. Okay, now you got the fast affines. Now you have to come here for the dereplication, chimera removal, generate OTU or ASV. Then you will get the count table, FASTA file and taxonomy, and you will get this kind of table, and you can do the rest of the analysis. Okay. So mostly some tools that provide whole workflow, Data2, which is present in R, USearch or research. It also can give you the OTU ASV. Motu is another thing, and Kind2 that provides a multi-interface environment that employs processing tools like those above. Infrastructure for easily documenting all core processes performed and interactive visualization. Okay, we will try to do this thing today. Okay, in our practical session. So Data2, which is a very interesting thing. Okay, so it can kind of resolute the data in a single nucleotide level. Okay. It can resolute the data in a single nucleotide level. Okay. The motivation, why lingering problem, what are the problems with OTU? False positives that I told you. I mean, thousands of OTUs when only tens of sequences present. Consequently, richness appear to be dependent on the library size, because library error will be incorporated in the OTU, and microbiome distance that appear to be dependent on the mostly the library size. Okay. So poor sequence and taxonomic resolution defined by arbitrary similarity radius, because you are taking 97%, okay, 99% radius. So that's kind of arbitrary. So what happens? Why 97%? Because people have seen that the 16S difference from one species to another species, there is a difference of at least 3%. But it happens for some species that 16S is different, I mean nearby like 99%. Okay, it's very close. It's 98%. Somewhere it is 93%. So it varies. It's not always stick to 97%. Okay, so that's the problem. If you just think 97 and 99%, you will miss many new things. Okay, you are clubbing together. So that's the problem here. So in OTU, and the accuracy, abundance estimates, biased and noisier than necessary. Too much noise will be there, because I told you by denoising you can remove all the noises. Okay, and the cost, poor data efficiency, larger cost to achieve the same inference, and computational cost is also pretty high for these things. Okay, and it's not so stable. So more you run, suppose same data, one time you run, you get 3200 OTUs. Next time you run, you get 3150 OTUs. So you can lose it. So people will ask you what is going on. Okay, so this thing happens actually. Okay, in kind 1 this thing happens. So the starting point for Data2 pipeline is a set of demultiplex FASTQ files. You have to take the demultiplex FASTQ files corresponding to the samples in your amplicon sequencing study. That is, Data2 expects there to be an individual FASTQ files for each sample. Okay, or 2 FASTQ files, one forward, one reverse. This is for paired end sequencing. If there is paired end sequencing, you will have 2 FASTQ files for each sample. It will be marked as R1 and R2 for Illumina paired end sequence. For others, you will get only one sequence file. Okay. So then demultiplexing is often performed at the sequence center. So you get by demultiplex data. In addition, Data2 expects that there are no non-biological bases in the sequence data. Non-biological bases means adapters, tags, those should not be there in the Data2 input. Okay, so your data should be just simply the biological data. Okay, And this may require pre-processing with outside software if, as is relatively common, your PCR primers were included, so you have to remove the PCR primers, tags and other things. Okay. So once the multiplex files without non-biological nucleotides are in hand, the data to pipeline proceeds as follows. First, it will filter and trim. It will do everything for you. Okay. It will filter and trim. It will give you the option what to filter, what to trim. It will create the quality map for you, what we did yesterday in first QC. It will create the quality map. Based upon that, you can say what will be the trimming trimming threshold or something like that, where you have to delete the 3 base pairs, 5 base pairs, 10 base pairs, so everything will be there. Then it will replicate the sequences, then it will learn the error rates. That's the beauty here. Okay, so the program is kind of learning program. For every data, it will learn its own error rates from the existing biological data. What it will do? It will take the larger chunk of sequences, okay, and think first that that's the proper biological sequence, okay. Then based upon that, it tries to learn the error rates for ATGC. Based upon that, it will create a table, ATGC, okay. Now, what will be the error rates from A to T, A to C, A to G, something like that. Now, based upon that, it will choose the next cluster and it modifies its error model. Okay, so based upon it goes on in that way and that way and that way, and it identifies the erroneous clusters or bases, and it will just remove those reads from the data. Okay, so the learning is very important here. It can learn the error model, error rate, and it is your sequence specific. Okay. And then infer the sample composition, then merge. Look, the merging is later. They do the merging very later on. Merge the paired end reads, make the sequence stable, and last remove the time errors. Then you can do the taxonomic assignment. Okay. So that taxonomic assignment, there are several algorithms. There are now machine learning algorithms for the taxonomic assignment also. Okay. So you first have to train that machine learning, like you have the database. Okay. So whatever database, you can give your own database of 16S sequences. And suppose you are doing V3, V4 sequences, then you have to cut your 16S database in V3, V4 parts. Okay, your own database. Then you can train your program, learning program based upon your own database. Then you give one of these sequences, and that learning model will predict what assignment should be there, and there will be a value like 0.8, 0.9, 1, 0.5. So more the number, more actually the matching you get. So this will happen totally in the learning based method. Okay, that's the beauty of this. Pretty fast and pretty reliable. So the output of the data to pipeline is a feature table of amplicon sequence variance. Look, it is called, it's not called OTU, it's called ASV, amplicon sequence variant. A matrix with rows correspond to the samples and columns to ASVs, in which the value of each entry is the number of times that ASV was observed in the sample. This table is analogous to the traditional OTU table, except at higher resolution, that is exact amplicon sequence variance rather than 97%, means it is just looking only one nucleotide condition and it is separate. So in one ASV, all the reads are identical. That's the beauty. Okay. So error rates are mostly learned by alternating between sample inference and error rate estimation until convergence. Additionally, a plot is generated. You can see those plots also, error rate plots, that shows the observed frequency of each transition, like A to C, A to T, something like that, as a function of the associated quality score and final estimated error rate. You can see, okay, and the initial input rates you can also see, and the expected error rates under the nominal definition of quality scores. That means it will learn the error rates on your own data. It will first estimate the quality, overall quality values of your data. If your data quality value is pretty good, its error rate will be different. If your data quality value is pretty bad, then the error rate will be totally different. Okay, so it will depend on your quality value. That's why you should ask more than 80% of data of having quality value more than 30%. Okay, that is very much required for this. And in addition, a plot is generated, error plot, that shows the observed frequency of each transition. Like here, this is the paired input. Now it's the quality profile, and then trimming, then unzip the collection, and it will do the error rating separately for forward and reverse. Okay, if you have single read, then it will go in that way. But you can see, yesterday we have seen that forward reads are pretty good, but the reverse reads are not so good. So it learns error rates separately for forward reads and for reverse reads. That's why they are doing it separately. Then they merge it and then they go for the taxonomic assignment and other things. Okay. So this is the overall kind 2 data is implemented here. Like you take the raw sequences, then demultiplex these, then demultiplex its sequences. You can join pairs here, then demultiplex sequences. Then you use denoiser a cluster. For the cluster you can use these UPers and other things. And here you can, for denoise, you can use data2 and other denoiser program. And then what you will get feature table. What is the feature table? Like OTU table. Okay. So here you will get column wise the sample name and row wise the OTU names. And this is called representative sequence. What is the representative sequence? So for each OTU which is representing a cluster, its representative sequence will be there. Okay. But for 97% you can see representative sequence will be more or less similar to others, but for ASBs, the representative sequences are totally unique. Okay. And then from the representative sequence, mind it, not from the feature table. From the representative sequence, you need to do the taxonomic assignment. Because from OTU numbers, you cannot do taxonomic assignment. For taxonomic assignment, you need these representative sequences. And you can do the taxonomy also from these representative sequences. You can do alignment, align sequence, do taxonomy and phylogeny. Okay. And from this feature table, what it will give? It will give the presence, absence and abundance. So feature table will give you presence, absence and abundance, whereas representative sequence will give you the taxonomy and the taxonomic assignment, the phylogeny and taxonomic assignment. Now you have to consider this taxonomy, assignment, phylogeny and feature table for to identify the differential abundance, bar plot, heat maps, diversity analysis, and then statistics and other plotting. So many statistics are there. So for this you need some statistical packages. Different R and other packages are there. You can use this. So this is mostly kind to give some default plots and other things which you can get by default. But if you want to do statistical packages in a nice way, like you can use the microbiome analyst also. So for the microbiome analyst, what you will need here? This feature table, this taxonomy table, and this fragment. That's all. Yes? We need to give the feature table. Now you can merge these things. Feature table and taxonomy can be merged and put in a single bar file. So you have seen that they are an option that whether the taxonomy is included or not. If it is included, then you don't have to upload the taxonomy file separately. If it is not included, then you need to upload the taxonomy file.
Feature table and taxonomy
separately. Okay. So those things are already there. So in case of microbiome analyst, you need this one, and this one. And another important thing you need. Can you remember? Metadata. Without that, you cannot do anything. Okay, so metadata is very much required. Okay, so this is the overview of kind 2. Now we enter into the diversity indices. Okay, so we learned about the alpha diversity, beta diversity, and all these things. So here we will just talk about the different diversity indices. Okay, so this is a presentation. You can see how many sites are there. 3, A, B, C. Okay. So here you can see how many types of trees are there, how many species. In A, 3, and B, and C, 2. So number of species 3, 3, 2, A, B, C. Now this is the alphabet, if you think in that. Just simply the diversity for 40%. Pretty simple. Now if you think, now I am coloring the trees separately. Now the species difference. Suppose you are considering A and B. Species difference is how much? 3 minus 2 plus 3 minus 2. How? Can you see? Huh?
Yes, what similar species are there, 2 are different.
Exactly. Exactly. So there is a 3, there is a 3. So this is matching, this is matching. This is unique, this is unique. So the difference will be 2. Okay. So from here to here, if you compare this thing, this is matching, this is matching. So difference will be 3, 2, 1. Yes? Unique sequences. Species. From here to here, this is matching, this is matching. So 1, 2, 3. So that's for the beta diversity. So whenever we are comparing between sites A, B, A, so that is called the beta diversity. And whenever we are calculating the diversity within a particular region, that's called alpha diversity. So this is, you can see the within group variation and between group variation. So this is suppose the gut microbiome. Indians, they have their own gut microbiome. You can see Europeans, they have separate, Americans, this is Africans, Chinese people, Australians, they have, you can see different gut composition of there. Okay. If you see within this Indian community, you will see again differences based upon their food habit, the culture, the climate, and other things. You will see the diversity. So you can think, if you are comparing between different continents, then you can think of this as alpha diversity, and this by this, this by this, this by this as a beta diversity. But whenever you are focusing on India, you are comparing east to west, north to south, something like that, then again it comes a beta diversity. So what we found in case of bacterial diversity increases from carnivory to omnivory to herbivory. Look, for the carnivores, the diversity is pretty less. And this is omnivores, diversity is in medium. But for the herbivores, diversity is. So being vegetarian, we will have huge gut diversity. Yes? So omnivores, you see, they are in between. Not total carnivores, not total herbivores or something like that. And you can see this composition change in the gut microbiome during African egg diversification. So we all know the Homo sapiens actually emerged mostly from the Africa, out of Africa hypothesis there. So if we consider this, you can see gorilla, pan, and chimpanzee. Okay. So you can see if we take the gorilla as a common ancestor, and people have identified the gut microbiome of these organisms. Okay. And they found there is some difference of 7 to 30 million years, and they found some similar organisms are present, but some are lost organisms, okay, which were actually present. So based upon the habitat, okay, the hygiene nature and other things. So you can see shifts in the relative abundance of microbial genera within the gut microbiome were inferred from each branch of the host phylogen. Okay. So genera whose relative abundance increase or decrease are listed. These are increased, these are decreased. Okay. So mostly we lost these things and we gained these things. Okay. From this common ancestry. So so we just did all these things by calculating all these diversity. So now we'll go into in deep in the diversities. Okay. So species diversity is a measure of biological diversity in a specific ecological community. Okay. It considers three important ecological concepts: species richness, species abundance, and species evenness. So the first thing is species richness, second is species abundance, and species evenness. Okay, in any microbial community. So species richness is defined as the number of species present in a particular area. It's pretty simple. Okay, more number of species present in area, a rich region. Okay, it's pretty simple. And abundance is the number of individuals per species present in this area. How much a particular species is present in a particular area.
Abundance, sir, you are talking in a particular species, there are number of individuals.
And on other hand, the distribution or the evenness. Evenness means whether all the species are present in same amount. If they are present, all of them are present in same amount, that's the even. But how even this is? Some are present in high abundance, some are present in very low abundance. So the evenness is okay. We just correlate these things with healthiness. In case of human also, okay. In different body parts, we used to say more species diversity is good, only except one region, that's the vagina. Vagina, there is a diversity is pretty less, only lactobacillus is present, and that's a good thing. Okay. So healthiness of an ecosystem, since the existence of a diverse and balanced number of species maintains the balance of an ecosystem. So that's the important thing. It's not the high diversity or something like that. Whether the eubiosis is there or not, whether the proper balance is there or not, that's the thing. Okay. So why we tell that higher diversity is good? Because we studied and found that we considered the gut microbiome of say hunter-gatherers, okay, then farmers, and then city dwellers, and we found that gradually the gut diversity is decreasing day by day. Okay, what does it mean? That hunter-gatherers are more healthy, we are more not so healthy. But in the other hand, if you see the food and the lifestyle is totally different. So you have to consider these things also. Okay. So a more diverse ecosystem tends to be more productive and has a greater ability to withstand environmental stress. It happens in case of environments. More diversity is pretty good actually. Also an ecosystem with greater species richness has higher productivity, making it more sustainable and stable and could respond to more catastrophes. Okay. So these are the species richness estimators. Which are these? You can see. One is first is OTU richness, a simply count of different species on OTUs. Okay. And observed species, this is simply count of unique OTUs in each sample. And then Chow index is estimated diversity from abundance data, importance of the rare OTUs. Here you will get the rare OTUs. And AC index, abundance based convergent estimation index, looks for the rare data. So these alpha diversity indices are for species richness, mind it. We used to call all of them as alpha diversity, but that's not true. Okay, each of the indices have its own importance, mind it. Okay, someone is giving the richness, someone is giving the richness as an evenness estimation, like Shannon-Wiener, Shannon-Weaver, Shannon entropy, inverse Simpson, Gini inverse, Barger-Parker index. This is for richness and evenness estimator. And phylogenetic richness estimator, that's a different thing. That's the phylogenetic distance, PD. We call it PD. It will consider that for this you have to give the phylogenetic tree. If you don't have the phylogenetic tree, you cannot estimate this thing. Okay, and it takes into consideration of the phylogeny of the microbe to estimate the diversity across a tree. We will go in detail about all these things. Okay. So measure of biodiversity, if you think again, two important measures now we have seen regards to the spatial scale are alpha and beta. The definition you can see in this way: alpha diversity is the average species diversity in a particular area or habitat, and is also termed as the local diversity. Okay, this is for exams actually. And it is measure of microbiome diversity applicable to a single sample, might be, to a single sample. On the other hand, beta diversity is a ratio between the alpha diversity and regional diversity. Okay, so it is the diversity of species between two habitats of the measure of similarity or dissimilarity of two regions. Okay. So alpha diversity gives an overview of the structure of an ecological community with respect to its species richness, evenness, or both. In microbial ecology, a common initial approach to assess the difference between environments is through the analysis of alpha diversity of the amplicon sequencing data. Let me tell you, from 2010 onwards up to this time, all the microbiome study they have included either of these indices, alpha diversity or beta diversity. This is a kind of prerequisite thing. Nowadays, these things are going into the supplementary table. No more in the main text. Okay, these are going into the supplementary table. But beforehand, these things were the main features in any report. But nowadays, these are not main things. But you have to do these things. You have to look into these things. Okay. So the alpha diversity again, you know, beta diversity. Another thing is a gamma diversity, measure of the overall diversity within a large region. It's a geographic scale species diversity. Okay, that's called the gamma diversity. So now you know alpha, how many species are in sample. It will give results is a single number representing the richness of the sample. Results are reported in a table or as a curve. Both you can see. Rarefaction curve you can give or you can give the table. Okay. Beta, which species are shared between the samples. Result is a matrix of whole sample similarities like A, B, C, D, A, B, C, D. 4 groups are there. Now if you can create the matrix and each matrix element will tell the similarity or dissimilarity between any two groups. Okay. And taxonomic, typically a single, sometimes multiple taxonomic concepts are present. Sequences are converted to taxonomic classification, and taxonomic classification are presumed to reflect evolutionary history. Our functional means whenever you are considering the whole thing. Okay, typically all other genes, not the marker genes. Only taxonomy we only do with the marker genes. But here you have to consider the all other genes, functional genes here. Okay. Now look, there is a 1, 2, 3, 4. 4 different regions are there. Okay. So here how many different species are there? 1, 2, 3, 4, 5. And here you can see 1, 2. And here you can see 1, 2, 3, 4. And here you can see 1, 2, 3, 4. This is fine. This is different. Okay. Now look, these are abundance. Okay. So they are present in phone number. They are 2. This is 1, 1, 1. Look, this is present like 8 or 10, something like that. It is only 1. So these things are there. Okay. Now this is the increasing richness and abundance. This is the richness is pretty less. But this is the highly rich sample in the even sample. Because all the samples are present almost in clear. Okay. So now the rarefaction is another important thing. What is rarefaction? So the rarefaction method lets you compare the number of species found in two regions when the sampling effort differ. So let me tell you, nowadays nobody is actually doing this rarefaction because you will lose data by rarefaction. Because nowadays sequencing depth is no doubt pretty high, and now the reviewers will tell you don't go for rarefaction, just do with the normalist form. Okay, because after rarefaction is simply the subsampling. Okay, just you are keeping subsampling from your sample, and each subsampling you are looking when the threshold is reached a plateau region. That's the thing. But you will lost in this way. You will lost most of your data. That's not nowadays is people are doing. Okay. But still we need to know this thing. The rarefaction method lets you compare the number of species found in two regions when the sampling effort differed. Okay, you are sampling from soil. Say you are taking a huge chunk of soil, but somewhere else you are taking a very minute amount of soil. So may not be you are taking the whole community there. So these things differ. So that thing you can actually manage with this rarefaction. So you would expect that greater sampling effort would yield a larger sample and more species. So you can't just compare the number of species found in each region. I mean, if you just take a 10 gram of soil from here and you take 10 kilo of soil from there, obviously you expect to get more species out of that. Okay. So rarefaction uses the data from the larger sample to answer the question, how many species would have been found in a smaller sample? Find it in a smaller sample. It's a subsampling thing. If you found n organisms in the less sample region, rarefaction takes hypothetical subsamples of n organisms from the more sample regions. Suppose you have 3 samples. Okay, in one sample you only get 20 species. In another sample you get 200 species. In another sample you get 2000 species. So in all these samples, ultimately you will take the 20 samples from all the species. You can take the all 20 from sample 1. Okay, and you can, by subsampling, you can take 20 from 200 species. And in the sample where the 2000 species is there, again you will do the subsampling and take 20 species. And then you can compare. Okay. So this average can be compared to the number of species actually found in the less sample region. The method computes a variance and distribution to help you judge how significant any difference is, to know the diversity. Okay. So the statistical technique or method used to evaluate species richness from the results of sampling is rarefaction. This technique is often applied to OTUs and is very useful in pollution and mostly evolutionary ecology. Rarefaction can be used to determine whether a specific sample has been sufficiently sequenced to represent its identity. Okay. This can also be used to infer whether a group of samples are from the same community or not. Okay. And the quantification of richness using a sample either measured as the number of observed species or approximated by the estimation is a common first step in microbiome studies, like this thing. So these are, you can see different samples. Okay. So you did subsampling here, but even after subsampling with higher sequence depth, you are getting more number of species there. Okay. But see, after this region, after say 10,000 or 20,000, you will get almost all the species present in any sample. So this is the enough thing. You don't have to go for this depth. So this is the point where you are considering all the samples. This is also true for here. For this is the kind of region. These two common patterns in rarefaction curves. Panel A shows a smooth incline in observed richness, panel B shows a steep incline and early plateau for all the samples. So for high depth sequencing, you will get this kind of things. Okay. So it involves the selection of certain number of samples which is either equal or less than to the number of samples, and then randomly discarding reads from the large samples until the number of remaining samples is equal to the threshold. That's the problem. You are discarding some reads. Okay. So nowadays it is not advisable to do. Okay. You are discarding many reads. You are losing many information from your data. Okay. So whatever. So this is a diversity, it is the influence of sequencing effort. Like this is sequencing depth, you can see these indices, Shannon, Inversinson, Barger-Parker. So these are mostly after this sequencing depth, it's fine. But the species richness, they need very high depth sequencing. In order to reach the plateau for the species richness, you need very high depth of sequencing, which is not true for Shannon and other things. Okay, so whenever you are doing species richness estimation, so people actually ask for this kind of curves. For others, they might not be interested. Okay, but we used to invariably give curves to all the indices. But that is not required. Okay. Now again this alpha diversity and the species richness we talked about. So we discuss here some of these diversities like Chow index, Simpson index, Shannon index, SE index, and the goods coverage index. So these indices we will now just discuss a bit. Okay. So can we have a break?
Yeah, sure.
So 5 minutes.
Tea time. We'll have tea and
So attendance is less today.
today is Sunday. And also the SLET exam is going on.
Oh, slate
So
is
some students they attended that. And here most of them are Christians, so they have
okay.
gone to our church.
This system is pretty nice. So this thing is kind of metastatic. Yes. Via, I mean, is there any industry code or something? At all? Yeah.
Solo hola.
That's big. What's this company? Yeah. That's what I thought. And it takes power from the oh, it's not so just try to find out the rocket. Oh, okay. They are going to that will be good. I don't know what So first start with the chow index. Okay, there's two types of chows are there, chow 1 and chow 2. Okay, and it's an estimated based on abundance. I mean, there are some estimators which is either based on the presence absence of species or abundance of the species. Okay, and another type is there which considers the periodic distance of the species. Okay. So here it is an estimator based on the abundance. Thus it requires data that refers to the abundance of individual samples belonging to a certain class, means certain grade. Okay. On the other hand, chow 2 is an estimator based on incidence, means presence absence. Look, there is a huge difference between chow 1 and chow 2. Thus it requires data that specifies the absence or presence of a species in a sample. So one is looking at the abundance and another is looking at the presence absence. Okay. So chow index is for estimation of species is given by this equation. So mostly we use this kind of alpha diversity matrices. I mean observed value by A and B to values they calculate from the data abundances. And then the Simpson index, we use chow index pretty much, and also the Simpson index. It's a kind of the degree of concentration when individuals are classified into types. Okay, like Edward Simpson developed it, it's equal to the probability of the two entities taken at random the data set of interest represents the same type. Okay, so it's again it's taking the probability of presence of particular microbes in a particular sample, and based upon that we used to calculate these things. And we mostly use this Shannon index. It's a pretty known index. It's an entropy kind of thing. We also call it Shannon entropy. Okay, it's pretty famous thing. It's called the Shannon diversity index or Shannon entropy. It's one of the most popular diversity indices in the ecological literature. Everywhere you will get to know this entropy. Means it will give you the enthalpy part mostly. The idea of this matrix is that more different letters present and the more equal their proportional abundance in the stream of interest, the more difficult it is to predict which letter will be the next one in the stream. Okay, it came from a physics thing. Okay, it's not mostly developed in biology. So it will calculate the probability of the microbial population. Okay, it's the enthalpy is calculated in that way. So it will take if the more kind of different organisms are present in different abundance, then the you can see entropy will be pretty high. The state of disorderness will be pretty high. And when the things are pretty ordered, the values are pretty less. Okay, so that's the kind of thing here. We'll calculate this thing also. So the AC index is another index is kind of
negative value. What is representing here? Negative value.
It's negative value. Its value is H value is negative. Okay, so it's just an equation. Okay, it's just an equation. Like we saw in case of rate quality score, we saw it minus 10 into. Can you remember that thing? Log 10 to the base t. Okay, so that's just an expression. Okay. So like this, the AC index is there that involves an arbitrary abundance threshold value, and the S abundance as the number of abundant taxa, and S rare as the number of rare taxa. So it looks for both abundant taxa as well as for the rare taxa. So let me tell you, some of these indices they actually consider only the abundant taxa, and they used to discard the rare taxa. And some indices are there which actually emphasize on the presence of the rare taxa. And that's, I mean, based upon your objective, if you can find the difference in the abundant taxa, then obviously you will not look for the rare taxa. But suppose you are not getting difference in the abundant taxa, then you go for the rare taxa. Okay. Similarly, goods coverage index is another one. This uses alpha diversity matrix in microbial ecology. F1 is the number of singleton OTUs. Simple, very simple, and N is the total number of individuals in the sum of abundances of all OTUs. Okay, so it will vary F1, F2 in this way. So now look at it. Okay, so again I have 1, 2, 3 samples. Okay, in this sample you have 1, 2, 3, 4 red and 2 blue. And here we have 3 red, 3 green, 2 blues. And here 1 red, 2 blue, 2 green, and this is another color. It's deep blue or something. So now we calculate the Shannon diversity index formula. What is the Shannon mathematically? This is the equation. Okay, is the p value. Where H is the Shannon diversity index, Pi is the proportion of individuals of ith species in the whole community, and Pi equals to small n by capital N. Okay, here for this community small n is 2, capital N is 6. Here for this small n is 4, total is capital N is 6. Okay. So where n is the individuals of a given type of species, capital N is the total number of individuals in the community, sum is the total sum value, and log you know is a natural log. Okay, it either can be base 10 or base 2. Okay. So now in case of rate, okay, now this is the OTU table I have created. This is sample 1, sample 2, sample 3. This is OTU 1 which is red, OTU 2 which is blue, OTU 3 green, OTU 4 purple. Okay. Now look how to create the OTU table. So the sample 1 red 4, blue 2. For sample 2 red 3, blue 2 and green 3. And for sample 3 red 1, blue 2, green 2, purple 2. Fine. Now we have created the OTU table from this visualization. Clear? Now what we'll do? Now we'll calculate this. You can just simply use this formula. Pi value means here pi value will be 4 by 1 will be 4 by 6, another time will be 2 by 6. So pi will 1 will be 4 by 6 plus 2 by 6 2 by 6. Okay, 2 times, and this for this it will be 3 times, and for this it will be 4 times, because our formula is there. So summation is there. So if you look at these things in this way, if you calculate the sample diversity index, the value you see this and this. Okay. So now from this you can see the diversity is most in sample 3, and diversity is least in sample 1. It's clear actually.
So do we need to consider that minus symbol in front of?
Yeah, obviously. I mean, minus sample should be there. The log value, the minus sign will be outside. Yes. So that will be so multiple. It will be plus. Like what we did in case of red quality. So you can see its diversity is high. Even just looking at the samples, you can just say this is the high diverse and this is the less diverse. And now you see you are getting this. Now in the evenness, look, evenness is again it's higher here. Look, higher here. It's more or less similar. 0.97 is 0.98, but it's not even pattern. Because 2 4. And here see 3 3 2. So look, Shannon index may be high here. That doesn't mean that evenness is really high here. Evenness is high here. That's the difference. Okay. That's the necessary to understand the meaning of different indices. What it is giving. Okay. Here it is giving the diversity, considering the abundance value also. Okay. Presence, absence, and abundance. Now look at the richness, simple richness. Okay. The number of species again. In this case, the richness is high here, but evenness wise it is higher. Total number of individuals it varies. Average population size, look, it's pretty high here. Okay, so all these things you can play with all the parameters. Like you can take these things and you calculate all the parameters, and you will have a clear idea what is they are telling about the community. Okay. So now we go to another interesting thing. Okay. Now this is a sample. How many species is there? 2. One is purple, one is green. Now say this is the phylogenetic distance between them. This is a phylogenetic tree. So OTU 1 is here, OTU 2 is here. Clear? Now this diversity is really pretty high because you have to traverse a long path. Here, then here, then here, then here. Okay, but suppose OTU 1 is situated here. Then diversity, which is phylogenetic diversity is pretty less. So now you have another sample where these two things are there, but they are located in that way. Now evenness wise and otherwise all the indices will be similar. Okay, but look at the phylogenetic diversity. They are totally completely different. That means these two species are distantly apart, and if it is present here, they are very close to each other. So that's why PD or phylogenetic distance estimation is another important thing. Okay, this is a totally different kind of estimation, phylogenetic distance. So what I already told you. Okay, if the OTU1 is present here, then the diversity is pretty low. So the things will be different. So now we calculate this phylogenetic diversity or PD. Suppose we have this sample, okay, Pseudomonas aeruginosa, Pseudomonas argentinensis, Pseudomonas flavensis. This is sample. Look to Pseudomonas monasteria. Here in sample you have Pseudomonas giardia, methanolipidata. You can easily say all of them are 3 samples, 3 different species. Okay, all the indices will be similar. Evenness, richness, Shannon index, all will be same. But you just looking at it, you can see phyletic diversity is high here. It's pretty low. It's medium, in between. Isn't it?
Yes, sir.
Okay. Now how to estimate this thing? Now we have this phyletic tree beforehand with us. Now what we do? We'll just simply place these organisms in the tree. Okay, suppose in this tree we calculate the PD, phyletic distance. Okay, all the pseudomonas will come together in one branch. You can just simply calculate this. Now in sample B, what will happen? These things are here with Escherichia coli. You just calculate the value. Escherichia coli is very closely related to the
Branch
pseudomonas.
distance, branch length we are calculating here.
Just you calculate the branch length. But in case of sample C, look, one pseudomonas, one giardia, one methanobibliobacter, and look at the PD. It's way higher, 0.95. Previously it was 4.35. So now the PD you have, if you just consider the PD here, if you can implement all the Shannon, all the Simpson, all the evenness, richness, will get identical data. And what you have to say in the report? I did not get any differences. Okay? Yes? But whenever you apply this PD, then you go to your life, look, I got a difference. Look, I got a difference. Okay, and your guide is in the happiest person. Okay, my actual happiness is what? Look how brilliant I am. Okay, but if you give the previous data, there you go. So, why I am telling you all these things? Because you have to look into your data and you have to understand all the indices properly so that you can use it in a proper way. That's what I am telling you. Okay. So you have to briefly go through all these indices, what they are telling you. Because so many literatures are there about these indices. Just I am telling you just one liner. It's like just one liner to you. I mean I am just not describing all these things in detail. Because if I try to describe a particular thing, it will take 1 hour. Okay. So I am just briefly telling you. Now if you build interest into it, then go and study by yourself. And you will get to know in detail what is going on better than me. Okay. I am just telling you very briefly the basics of all these things. Like PD, we always tell others to use this PD thing. Just do one evenness, one richness, and Shannon and this PD. Okay, so do these things first. Then see what is going on. Okay. So the conclusion, what will be the conclusion? Sample C is more diverse than B, which is more diverse than sample A. Okay. It's pretty easy thing to understand. Now, the beta diversity. Beta diversity is diversity between
Samples.
not within the samples, between two groups, between different samples. So it will tell you the variation of microbial communities between samples, how different is the microbial composition in one environment compared for others. So yesterday we learned about the pangenome in comparative genomics. And what we did? We first looked for the common part, mosaic part, unique part. So more common part means more similar, more less common part means less similar. Same thing we will be doing here. We will take two samples and we look for how common they are in terms of presence absence, in terms of phyletic distance, in terms of abundance. These three things are there only. One is presence absence, one is abundance, and one is phyletic distance. Okay, so we will look in the beta diversity how the things are kind of different between the multiple communities from different environment. Main focus is on the difference in taxonomic awareness profile from different samples. So the break rate is dissimilarity. That's one of the most important thing. We always try to first do break rate is Jaccard and other different matrices are also there. In Baker-Tees, it is based on the abundance or read count data. Mind you, it's based on abundance or read count data. Differences in microbial abundance between two samples. You can do it at the species level, you can do it at the genus level, you can do it at the OTU level. Okay, so the first level is phylum level, then phyla, then family, class, order, all these things will come. At the last, you can do up to at OTU level or ASB level. That's the last thing. Okay, so you can do it at the species level or OTU level or genus level, whatever you want. And its value varies from 0 to 1. 0 means the samples share the same species and they are identical. So the value is 0, so they are identical. 1 means both samples have complete different species abundance. Mind it, here I am telling this as a dissimilarity. If I tell this Baker-Tees similarity, then 0 means complete, totally different. 1 means complete. Here it is dissimilarity. Dissimilarity means higher the value, higher the dissimilarity. Clear? 1 is the maximum value, so 1 means it's completely different species. Okay. So Jacob distance is there again based on the look. This is the based on the presence absence of species. Does not include abundance information. So Baker-Tees it is including the abundance or read count. So I told you abundance one important thing, presence absence another important thing, phyletic distance another. Yes. Please try to remember. In all the indices these are the pretty important things. So the Jacob distance is based upon the presence absence of species and it does not include the abundance information. Again, difference in microbial composition between two samples. 0 means they share exact the same species. 1 means both samples have no species in common. Presence absence means we are just comparing. How about that tree figure? Can you remember the tree figure? We were just comparing the similar trees present in two communities. Okay. Same thing is here. 1 means both samples have no species in common. All the species are different in two different samples. And another very powerful indices is there for the beta diversity is called the unifrac. Okay. So mostly here you have to keep the phylogenetic tree like what we did in case of PD. Okay. So it will take the sequence distances. Okay, it means phylogenetic distances. Based on the fraction of branch length that is shared between two samples or unique to one or the other sample. So simply it is considering the branch lengths. How the OTUs are close to one another or different from one another based upon the branch length. So two different unifrac techniques are there. One is unweighted unifrac, one is weighted unifrac. What is unweighted unifrac? Purely based on sequence distance, does not include abundance information. So simply based on the phylogenetic tree or simply the sequence level similarity or dissimilarity, if you find out the distance, that is called unweighted unifrac. But if branch lengths are weighted by relative abundance, okay, branch lengths, if you just weight them, suppose one particular OTU has huge abundance, okay, you give them more weightage at that branch, then that will be called the weighted unifrac. So here with the branch length you are considering the abundance information also. Okay, so in weighted unifrac you are considering both branch distances, okay, you are comparing the sequences and as well as the abundance of each of the OTUs. That's the difference between weighted unifrac
Weighted, unweighted is summation of all the branch length only.
Yeah, it looks for the how similar those things are and how dissimilar in branch length are. So then, now this is the differences. The next important thing is whenever you are calculating this difference between say two groups, the statistical test. You have to run F statistics or other things so that you can tell whatever distance you are getting whether that is significant or not. And you will consider P less than 0.05, but in microbiome for others nowadays P 0.05 is not acceptable. You have to go for the adjusted P value. Adjusted P value, which is called Wolfram correction. Okay. So means all these like in profile, okay, you are just taking one by one abundance, bacteroids versus firmicutes versus thing. But you are doing this for say 100 different individual trials. Then you have to correct this error by dividing the number of trials. That's called the Wolfram correction. The different corrections are there. So that is nowadays people are looking for also. Okay. Corrected P values. So now here you can see beta diversity is phylogeny based assessment of difference in overall bacterial community composition. The unifrac distances are based on the fraction of branch length shared between two communities within a phylogenetic tree, constructed from the 16S RNA gene sequences from all communities being compared. I already told you about the unweighted and weighted. Okay, so now this is the visualization or ordination. Okay, this is another important concept, principal component analysis. Okay, so component analysis means suppose you have multiple data based upon some factors. So the component analysis will try to find out the eigenvectors, which is describing the major difference among the samples. Okay, axis 1 give you or P1 will give you the maximum difference present in the sample. Okay, then axis 2, then axis 3, then axis 4, then axis 5. Suppose here axis 1 is giving 80% of difference. That means this sample is totally different from these three samples. And here look, axis 2 is giving only 19% of difference. So these samples are obviously different from these, but the difference is not that great. Because here the percentage of variance is really high. Okay, it's describing 80% of it. So in this way difference is pretty high. Although these two are similar samples, these similar treatment, these are similar treatment, but still you can say the difference is high here.
That's how, because if it is from the same sample, if it, and actually in some data it's showing different position, not in the same cluster.
It may happen, these are biological samples.
Okay.
It is quite expected to happen this thing. If you get clear cluster here and one cluster here, that means there is some problem. That's true.
Yeah.
These are biological samples and the heterogeneity is the main theme of biology. Okay, so many things are going on. When you get, like in RNA, we get a distinct difference between gene expression profile. When you have a healthy cell RNA sequencing and one is kind of drug treated cell, then you will get a clear difference between them. Okay, like in cancer cells, when you mutated the P53 and in the third stage or fourth stage, okay, you will get a drastic difference in gene expression between normal cell versus that cell. If the difference is so high, then you can see. But otherwise, you hardly can see differences. Okay. Now,
otherwise you have to go for outgroup. Then you will get a difference.
You will get that, which is the outgroup and why it is like looking like a outgroup. And also here the problem is you have considered only 4 samples. It is not at all advisable to work with only 2 samples in each group. Then, what is the minimum value? At least 6 samples. At least 6 samples. More will be pretty good, but at least 6 samples.
per group.
for estimation of the different statistical parameters. Okay,
Yeah.
at least 6 samples is pretty good. But obviously should be more than 3. Should be more than 3. So this ordination is another important thing by which yesterday we did it in beta diversity. Okay, you have seen this nice plot, 3-dimensional plot. That was the kind of this ordination plot. Okay, it was showing nicely how these things are. So when 2-dimensional, you have seen only 2 axes, equal 1, equal 2. But in 3-dimensional, you have seen 3 axes, 1, 2, 3. If the 4-dimensional is possible, then you can see 1, 2, 3, 4. But it's not possible. Okay, but this diversity, I mean this variation decreases whenever you go to the other axes. Sometimes, suppose the first axis is giving 20% variation, second axis is giving 19% variation. Then you have to look for the third and fourth, because overall variation is only 40%. Out of 100, first 2 axes, if it is describing only 40%, then other axis is describing the 60%.
Yes.
There might be some problem. So look for other. But whenever these two are describing almost 100%, there is no need to go for
Other
other.
axis. Okay.
Clear? So you need to calculate this, you need to add this up, and you need to see whether it is describing the majority of the variation. Okay. So for here, like you see, 7 different colors, 1, 2, 3, 4, 5, 6, 7. 7 different colors are there in these samples. Now suppose this is a treatment 1, this is a treatment 2. Now what is the influence of treatment A and B? What is the species diversity along the transects and gradients? Species difference among the samples are positively correlated to beta diversity values and inversely correlated to similarity. So now you calculate the beta diversity considering these two things, and you can also do the phylogenetic analysis by unifrac method, and distance between samples reduced if phylogenetic approach is used. Okay, because here you can see this is located here. Now if it is located here, then obviously the phylogenetic distance will be pretty much reduced. Okay, now we will come to the Bragg-Curtis dissimilarity. Okay, so I mostly use this Bragg-Curtis dissimilarity. It's very much useful in the quantification the differences in species population between two different sites. It's used primarily in ecology and biology and can be calculated with the following formula. It's pretty easy formula. That's why I used to describe these things to all of you. Okay, so Bragg-Curtis Ij. Ij means any two sides. I is one side, J is another side. Okay, I and J are two sides, any two sides Ij. Equals to 1 minus 2 Cij divided by Si plus Ji. It's pretty simple equation. Look. Now, this Cij, what is it? Cij. Cij is the sum of only the lesser count for each species found in both sides. Means common species found in both sides. Okay? The common species found in both sides. And Si and is the total number of specimen counted on side 1I, and Sj is the total number of specimen counted on the side J. Suppose two samples are there, 10 each having the 10 species and all are identical to each other. Then it will be 1, 1, 1. Okay, so 1 minus 1 will be 0. They are all identical.
Yes.
Here, if these all identical and all having two samples having 10, 10, then this value will be 20. This will be 10 plus 10, 20, 20 by 20, 1, 1 minus 1, 0. So they are similar. Okay, and if this value goes very down, I mean pretty less Cij, then it will be close to 1. If the common part is pretty less, then this value will be pretty close to 1. Suppose Cij is 0, then what will happen? There is no common species between two sides. Then what will happen? What will be the value? If Cij is 0, then what will be the value of BC? 1 means highly dissimilar, no matching between two samples. Clear? Okay. So now I am showing you an example. Okay, suppose this is aquarium 1, this is another aquarium. Okay, this is a tank 1, you can see 6 goldfish. 1, 2, 3, 4. These are not looking like goldfish, but think these are goldfish. Okay. And 7 guppies. So these are again not looking like guppies, but think this blue color guppies. And this is like rainbow fish, although they are white. Okay, but still we call it rainbow fish. Okay. So when you excite these things with laser light, you can see the rainbow color. Yes. So here you can see in tank 2, 10 goldfish and 6 rainbow fish. Okay. So to calculate the break-curtis, let's first calculate the Cij, the sum of only the lesser count for each species found in the both sides. Goldfish are found on both sides. Okay. The lesser count is 6. Only 6 count. That's why it's called the lesser count for each species. Okay, on one side, so they can be added in here. Rainbow fish, though are on both, and that is index. Mind it, this is different from the dissimilarity. Okay, Baker's index, and to calculate it, you simply subtract the Baker's dissimilarity from 1, and then multiply it by 100. So index is kind of, yes, subtract the Baker's dissimilarity from 1. 1 minus Baker's dissimilarity, and then multiply with 100.
Like same like
It's kind of indexing. Yeah, you are doing the indexing. So this fish example, Baker's dissimilarity was 0.39, and if we wanted in terms of percentage, it's kind of 39%, but Baker's index will be 61%. So it's another easy way to identify Baker's indexes. If you just convert the Baker's dissimilarity in percentage, then you minus this thing from 100, and you get the Baker's index. Okay. So now you know that it's pretty easy thing to. So identical sites have a Baker's dissimilarity of 0%, and a Baker's index of 100%, because it's 1 minus. Okay. And sites which say no species would have a Baker's dissimilarity of 100% and Baker's index of 0. Okay. So to calculate the Baker's between two sites, you must assume this is the assumption used in calculating the Baker's dissimilarity. So there is assumption in every type of calculation. Okay. So I am just describing it in case of Baker's because this is easier to understand. So the calculate between two sides, you must assume that both sides are the same size. That's a very important. Okay. Either in area or volume. As is relevant to species count. This is because the equation doesn't include any notion of space. It works only with the counts themselves. So it's up to you. Okay. If you do it differentially, I mean nobody can help. That's why you need to understand all these things. Okay. So if the two sides are not of same size, you will need to adjust your counts before doing the Baker's calculation. Okay. Very good. And there are different ordination techniques. After data diversity, you calculate the diversity indices, the values. Based on that, you will now plot this thing. Like PQA is one of the very common principal component analysis, also called metric dimensional scaling. It's a kind of dissimilarity or distance matrix. So all these things. So from this, like here, we got this value. Okay, BCIJ value. So see, so here I have one sample, two sample. So I got this value. Now suppose I have 4 of these attributes. Then what will happen? Then we will create a matrix. Okay. And here 1, 2, 3, 4, 1, 2, 3, 4. 1, 2, 1, 0. 2, 2, 2, 0. 3, 2, 3, 0. 4, 2, 4, 0. Then 1, 2, 2 we will put this value. 1, 2, 3 we will put the value. 1, 2, 4 we will put. So we will create the dissimilarity matrix. So for any number of samples, we have just first created this matrix, either similarity matrix or dissimilarity matrix. Mostly the dissimilarity matrix in case of beta diversity. Okay. Now, whenever we have the matrix, then we have to analyze the matrix. And in the matrix, you are getting pairwise differences. All the pairwise differences are there in the matrix. Clear? Then we will do this kind of coordination techniques. Okay. Either you can use this one, either you can use double principal coordinate analysis, it is a two-step PCOA, or you can simply do the principal component analysis. It is a coordinate, it is a component analysis. Is a preserve the variance of the sample, in particular HPCI is an hybrid method, where taxa abundance is combined with a file unit to create a contrast that are used as input variables. So these things you can later on use. So now there is another
For
different
that,
thing.
sir, we have to use which data, sir, for this PCOA, this coordinates data?
The matrix.
Like
The difference you are getting.
no, sir, the data after generate, after I'm telling about data, sir. Which one I have to plot? We have to plot there. The PCO, suppose I am creating the kind of PCO. After that, I am getting that file, the number.
No, you after using the beta diversity, now
So
you
many
got the
files.
matrix.
Yeah, diversity
Okay.
matrix.
From the matrix, the program will generate the different ordination techniques, ordination pictures. NMDS is there, PCA is there, PQI is there. So they will show in a three-dimensional or two-dimensional space how each sample is located based upon their relative distances. Okay, they will calculate the in fast axis wise. They will calculate the maximum dispersed kind of samples. And if you found your group, group 1, group 2, they are highly separated in first axis wise or second axis wise, then you can say there is a difference. And then there will be statistical test. So in unifrac, as I told you, it will consider the phylogenetic distances. Okay. So there actually kind of it will again create the distance matrix from the phylogenetic distances. So the unweighted unifrac will create the phylogenetic this kind of distance matrix from looking at the overlapping of the phylogenetic positions between any two samples. Okay, species present in any two samples. Whenever you are adding the abundance values or weightage, you are giving the weightage to the edges based on the abundance values, then that will be the weighted unifrac. Okay, it's a bit tricky, but I mean, you can just here you can see the red versus yellow, the red versus blue, then yellow versus blue. These are the different samples, and then you create the distance matrix. Then either you can represent this as in the PCOA or in the hierarchical clustering. Okay, this is the unweighted unifrac. Okay, we are not giving the importance of abundance of each of these species. Okay, this is unweighted unifrac. Weighted unifrac means you are also considering the counts or abundance of all these OTUs. Okay, so this is the weighted unifrac, which kind of weighs the branch length by the abundance of bugs, means OTUs. Unweighted only uses the presence absence. So these are the references. Okay, these are the kind of different papers and the things I considered for these particular informations. Okay, and I think that's all. Okay, so now we go for the practical because we need more time to
Yes.
okay.
So you have already done data set? You have data set?
Yeah.
You have to.
Oh, one another thing. My paper is there. Oh, okay, yeah. So I have that kind to protocol. So how to? I think I can project it there itself. Yeah.
So I think better we one time you explain here like or
Yes, sir, this will save.
No, that time to protocol means how to just we have to install that,
okay.
and then we have to follow the protocols. Okay. I mean, what we have to do. There are, I mean, what you have to do that.
Like what are that? Like click on down.
What are the commands?
See here, see in the projector. This icon, okay? This is our Ubuntu. This is
Dear participant, please calm and quiet because listen the instructions clearly.
so in every setup we'll see this icon, okay? Down here in taskbar. This is your Ubuntu. That is your Linux operating system.
Shall we open?
Wait, just I'm telling you, later you have to use that only.
Please note down, okay? Step by step, okay?
Ubuntu, why you are using?
This is, yeah, that is Linux because we are using this all our Windows system. So nowadays we can use in Windows also, we can use the Linux system. For them we have to install this software.
We can only get to know micro, not the keys. So first of all, could you please show how to just install that?
Yes,
You can
sir.
then please rather.
So to install Ubuntu, what we have to do, we have to.
G1 because the screen is not visible.
You see there? Yeah, you see.
So everyone is ready?
Yes.
Yeah. So this is our Windows system, okay? So to install Linux operating system in Windows, so what we have to do, we have to install one software called Ubuntu. So where you will get this one? You will get this software in your Microsoft, Microsoft Store. So what you have to do? You have to go to Microsoft Store.
Microsoft Store.
Yeah.
Note down. Microsoft Store. You have to go there.
And then here you have to search for Ubuntu.
Ubuntu. So then you type Ubuntu.
Ubuntu.
Ubuntu. Note down.
Yeah. Then you have to click on get.
It won't.
Yeah.
After that you click get.
For our system we have downloaded this one, Ubuntu 20.04.5 LTS.
This is I think the latest.
Yeah, latest one.
You please note that.
So, 22 has.
This may vary time to
It is
time.
okay, yeah, 22 has come.
But I think it's a beta version.
Yes.
Okay.
It's
So
not
20.04.
Compatible.
Yes.
0.5 LTS.
This is compatible.
Okay, all noted down. Then you can open.
Yeah.
Open.
Not open, don't open now. You have to download. You will get the option get. Then you have to click on get and it will download in your system. After that, what you have to do? You have to go to search again.
Where you are getting?
It's already downloaded, sir. It's already downloaded for all the
See
system.
already down.
Before that, if I want to download what
We have to click on get, sir.
Oh, see.
Get and
Screen
then
you will get instead of open.
Get.
You will get get.
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 Yeah. And then it will download and then you have to install it.
Install.
Just like the other software how you are
Please-
doing in your
Please note
laptop.
that link.
Okay.
Get after that install.
Yeah. And then after that what you have to do? You have to,
Let them install first.
so it's already installed.
Oh, it's already installed.
Installed in all the
system.
system, yeah.
Oh, that's
They
great.
can install in their laptop later, I will
That's
show
fine.
them.
That's fine.
Testing you tell, then they will do it themselves.
Yes.
Okay.
And then what you have to do, as this is a Windows system I have told, so what you have to do, you have to search there, turn Windows feature on or off. Write this. Turn Windows.
Where you have to go?
Yeah, you have to go just search option in the laptop.
Okay, search option.
Yeah.
You know all search
Yes,
option.
yes.
In search
After
option.
installing it, we need to do this.
Yes,
Yeah.
you have to go to search option, then you have to see the turn Windows features on or off.
And if you have any confusion, please contact.
Contact me.
Okay?
So you have to go there. So here you will get so many options what you have to turn on or off. So here what you have to do, this one, Windows subsystem for Linux.
1 minute.
You have
Windows,
yeah,
Windows subsystem for Linux. That thing you have to
because it's it's a
put a click and
yeah. Normally it is like this.
Okay.
You have to tick it, and then you click on OK. And then the system will say like restart it. So you have to restart it.
Restart the system.
Yes. That's all.
Other place? Noted down? Good.
Yes, sir.
Yes. Right.
So this is how you are operating the Linux system in your Windows platform.
Okay.
And then you have to download the KIM2 in the Linux system.
My suggestion is, if you want to do bioinformatics a lot, then just format this thing and install the Ubuntu.
Ubuntu.
Okay, that's totally free. Okay, and you can not only this kind to, you can do any. Lots of options are there, like Philosophic and lots of options.
Ubuntu.
So if you are really interested to do the bioinformatics, you should learn the Linux first. The shell scripting you should learn. So lots of books are available, small books are available.
Already
So
we have sent one book, no? Yes, our bioinformatics person sent. So
So-
go through that.
That's very important. If you want to do, the continuing this NGS in later on, like in Smart, if you do want to do this RNA sequencing analysis and other things. So for everything, you have free packages in Unix itself.
Then after that, so now system, it is everything as downloaded. And after that, how to start?
Yes.
Wow.
Wait for something. And I can show the command, na? What I use.
Dinata,
Yes.
Now it's-
one query. Like in Windows 11, there is the option. But if somebody has Windows 10 or something, there is no Windows subsystem.
Dual
So what?
booting.
Dual booting have to do. VirtualBox also you can do.
VirtualBox.
VirtualBox is another option.
VirtualBox se kar sakte hain.
VirtualBox, but in VirtualBox you will have too many problems because
Yes.
these are the memory intensive.
So even some part is not supporting in the
So
Windows.
it's better you go for the dual booting.
So yeah.
Yeah, you can go for dual booting.
Specific.
So then we have to download the Miniconda. Miniconda is an environment like where the
So now that they have to do it now, here or you have
So
already.
done.
Oh.
That is
You
also
can
there.
tell that what is.
Yeah, these are the command I am showing now.
No, they have to do now, yeah.
wget. So wget is a command
You
in
You can open,
wget.
you can open your like this, yeah. For that what you have to do, yeah?
Where we have.
You have to-
Yeah,
Yeah, you, in your system you can open.
yeah, open and write just history.
One. How to open?
How to open, how to.
Just click there.
This one?
Yeah, click on your Ubuntu. One click.
Oh,
Yeah.
click on Ubuntu. It is coming?
Yeah.
Yes?
Yes.
Ah. Everyone? Right,
Okay.
next.
Then just type history.
Just after that, the
Yeah, just write history. So what we have done to install, don't give any space. Just write history.
Don't give space.
It's not. It's not.
Don't give space. Type history. That's all. After history, enter.
Yeah, just enter.
Yeah, enter.
Yeah, so what we have done, oh, this system we have run the, it will not come.
What about here?
He wrong, na sir, HISTORY.
Ah, CIA spelling mistake. Yeah, history.
Huh.
Small.
Yeah, all
TORY. TORY or history.
Because these things, this old system we have already used the things, na?
Ah,
So it's
enter.
okay.
Yes.
No. This is, we have run the, actually this system we have already used for analysis, that's why all the commands are coming.
That's great.
All the commands they got. This thing we haven't done. So sir, please explain the wget and all, sir. Yeah.
You can explain
No,
it.
sir,
na.
please
No,
explain. That's
It's
good, sir. I
better.
will
You-
also get.
Actually, if any mistake is there, he will correct you. So that way, yes. Yes. Ah.
I can give
No,
you one
here, no,
Linux.
here, no, here it is
I
not
have
coming.
that.
What you have to do?
Come, sir. done, sir, already.
That's all.
Yeah. History, we done.
History, we have nothing.
Oh, because this, no, like that.
Don't give space, only type history. No, you are giving space, don't give space. No, here.
Down, down, or like.
Down.
Oh, keep it something working.
Down, don't give space.
Just like.
That's all. Okay.
This is. Yes,
So this is the data, okay.
yes, sir. Done, no?
All are slow learners. You have to think all are slow learners. So slowly you have to take one by one. Okay. And then what I need delta. And two classifiers. I think one.
One of them.
Yeah. And one more.
So do you need this Linux comment?
Yes, sir, it's good, sir. I have to copy in everyone's system.
Here next what they have to do?
No, sir.
Get right.
Yeah.
No, how I will install my Windows is 7, 8, 9, 10 like that. So
We
please
have to go
highlight.
for dual mode, sir.
What is
Dual
that?
booting.
That's why I don't know, no. You have to tell.
Like they have to separate their
Hard disk.
hard disks to half portion for Windows or some like half data for Linux. They have to go for dual mode. Yeah, partition. We can say partition. Yeah.
Oh, yes, for that you need to again format.
Yeah, we have to
one
go to
particular-
computer fellows.
So you need to format.
No, what?
Dual booting mode. Dual booting mode.
Dual.
Dual. Double. Partitioning.
dual-
You have to do partition.
dual?
Booting.
Oh, dual booting here, not down here. If you are having Windows 7 or 10 like that,
No,
you have to go for dual booting. And contact the computer personnel, they will do dual booting, okay?
No, I don't know. It's
First you need to partition your this.
Partitioning.
Where am I now? Just open Microsoft.
okay, all of you. Please look here. Okay.
And then the Windows.
So there will be a short class for Linux for NGS. Okay. So you need to learn Linux for the NGS systems, NGS analysis. Okay. So the vast majority of bioinformatics programs are written from or can be easily be compiled for Unix-like OSs. Okay. So the Unix philosophy permeates the ecosystem. Things to know, command line environment, what you have, mental model of directory structure, and the program input and execution, and archives and compression. These things we can discuss. So this is, you look, anatomy of a command, any kind of anatomy, I mean the command you give. So you see this $sign, then CMD. So this $sign we call this prompt. Look it here, we call it prompt. And this CMD we call it command. Okay. Now these signs -a, these are the flags. Okay. This whole thing is parameters. Okay. This whole thing is parameters. And this is the command you can give. So for different utility we will give different commands. Okay. So and these are the flags. Each of these flags will tell you different things. -a is telling you different thing. -f is telling you different thing. -10 is different. T is different thing. And these are the parameter values. Okay. The value you give. And whenever you see -o, that means you are giving the option for output file. Okay. So -o outer.txt means you are suppose this running this particular command from a particular folder. In that folder you will get a output file out.txt. Okay, and -i means always the input file. Okay, here two input files I am considering, file1.txt, file2.txt, and this is suppose long form parameter. So you can mention these parameters here. So this thing you can do actually anatomy of a command. So echo means you are telling to print echo -e. Baby RNA says goo and /n baby DNA says gaga. Okay. So you can do it here. Do this line.
Type that line in your
command.
system.
Encode minus. Echo minus. That is
-e, then in double quote. So RNA always say goo u, okay, and /n \n baby DNA says gaga in single quote. Goo and gaga in single quote. Yeah, okay, now you remove the -e. You can use the up
Yes, sir.
arrow.
RNA says
You can use this up key. Okay, up key there and then remove -e. Let's see what happens. There is some mistake.
Oh, nothing coming here.
please
Oh,
repeat
cannot
it
now.
again.
You do it again.
Sir, now it is clearly over.
Huh? So the thing is
Done,
not.
na?
No, your command is correct. You check here.
Comment
Not-
only, wait. So there sync space is there, na?
Yeah, the space is there. So use the up arrow. Yeah, there will be a space after echo. Yeah, then there will be space after E. After E, yes. Now run it. Enter. Okay. So spaces are very important.
Spaces are
Very important.
Here also. No,
So
no.
here now you,
Enter.
after all of you have done it,
Done?
done.
Done, your one.
So all of you,
Yeah, done, sir.
you done it.
Space, you use space.
Okay.
Yes.
So no. So
No.
did it. Okay. So now you see most of you already did it. So just remove the -e.
Yes, sir. Removal also is coming. Baby says.
Remove minus.
You just use the up key actually.
Just E. Yeah.
Just D and
You-
So
again
if you remove the -e, then you can see this \n will not be interpreted as new line. So the -e, this flag is actually telling this \n as the new line. So you are putting new line here. It is coming in a new line. Whenever this \e is not there, this flag is not there, so it will be right in a same line. Okay. So here this echo is the command. Echo is telling
Minus
print
E,
it in
minus
the screen. Okay. So echo is the command, E is the parameter, and here you are just telling other things to print here. So this is the string to standard output. Now this is another very important command, sudo. Okay, don't do it. Okay. So sudo is kind of a very powerful command. Execute a command as another user. Suppose you are in guest user, and if you do sudo, you are asking for the administrative password. Okay, administrative access. Now if you know the password, then you can go to the administrative mode and you can do anything. So here minus space then rm, it will remove file or directory. Okay, and R minus RF means remove directories and their contents recursively, and this is actually you are forcing it. Okay, so if you run this thing and if you know the administrative password, what will happen? It will erase everything in your system. So don't do it. Okay, so that is why it is necessary to understand the meaning of each and every command. Don't do it blindly. You need to understand what is going on here. Okay, otherwise if someone gives you these kinds of command and you just do it, then everything will be gone. Okay, so another very important thing is path. Okay, it's like address. So if you see in here only, suppose this one, okay, in desktop. Okay, if you double click it, you see here desktop. Okay, but if you click here, look, the path is given. C : users, then Acer, then desktop, then desktop. So C will the desktop under desktop. Okay, so if you go here, look, you just click here, you will see the whole path. So this is the address of any particular file or any particular folder. Suppose if you go here in baby, I mean this say desktop the smiles. So what will be its address? Its address will be copy path. If you can copy path, and if you put it in notepad, then you can see. Can you see it? Yes. So this is the address of this particular file. So it is like this is district, this is state, this is country, this is art, I mean our planet, this is universe, something like that. You can think in that way. So every file that has a defined path, okay, where it is located, its address is given in everywhere. It's irrespective of Windows or Linux or anything that has a defined path. All the files. That's very important to understand, and that's what you see here. This is showing a path. Okay, $sign, command prompt, then it's under variable, then library, then flatpak, then export, then share. So this is absolute path. You are telling all these things in detail. But these are the kind of dot. If you put dot relative path, it will go its upper state, it will go to its upper state. So first you need to understand this one only. Okay. Next, look at this path. Okay, in Linux, everything will be under the root. Okay, there will be the root directory, that's the main directory. So only the kind of administrators have this access, this root access with the administrative password. Then you have different users. Okay, suppose there is a max folder in the users, so its path will be dash, means it's root there, then users, then max. Similarly, see, after the root there will be bin, etc., users, temp, user, these are the folders. And say in users there is a max folder, there is a min folder, there is a work under this max, and there is a work and play under this min folder. Now, this is another important concept, input output redirection. Okay, whenever you see stdin or this arrow, that means it is telling read from standard input. What is the standard input? In computer, standard input is this clicks and this keyboard. Okay, that's the standard input. And here standard input std out means write standard out to a file. Okay, so look here, cat is a command which can concatenate. You are telling two files, list A and list B. So it will concatenate these things present in file A and file B to a new file, file C. Now you can do cat, just put it in a list.txt. That's a new file. Okay. So this is the stdin. This will be the read from standard in. Stdin, if you do the double quote, then it will append. Means it will not create a new file. It will append. Means it will add the new things to the existing file. Okay. And this will give you the standard error to a file. Okay. You can do these things later on with yourself. So the like who means where the machine is. You can use this command again here. Okay, who, and you can see how many folders are there, folder names. So this sign lets you read from and write to files what if you want to pass out to another program? Okay, like who will give you these all information? Okay, it will print all this information. So you type who here, just type who, WHO. You go to any day, I mean, huh? Any directory folder. Go to any directory or folder where you have, just do cd. Let me see whether there is anything or not.
Sir, something wrong.
Hmm?
I think it's something wrong.
How?
You can't like this.
No, you are not, this,
Yeah.
just escape. Escape. No,
No,
escape button, press escape.
See, I have sent one directory information file like, right, in WhatsApp group. Yeah, please, according to our system, write that directory. I have sent one, yeah, file information.
Again, you send it.
Yeah.
So just cut it and start it again.
First
System information, I'm sending.
System information first, yeah. You
I
can.
have tried all this.
Are you getting these things here?
Yes, sir.
Just try that first.
Since-
Okay, now you check it here. That all system information you load it as per your system. What is the number?
Number 7, yes. 6.
It cannot.
So we have to write in a note.
So this file name is not good.
Yes,
Here we have to
Because
write.
you have used space.
What is your system number?
Folder I have the new station.
Number 8.
System number.
System
See,
number is
05.
8.
System number 8. Then you have to see the system number.
Okay.
What your rest of so accordingly you do that. System 1, 2, 3, 4, 5, 6, 7, 8.
Just write city everything, na? What system number 11.
You want to use
What I don't
open
have
that one, no?
Okay, so I have to type this.
Yeah, type that one. Let's type CG first. Like that. You can type that one.
Again, starting from C.
Just put enter. Okay.
Again, I should write from C.
Space.
Not just do it WHO. Why it's not showing? Maybe it's, those passwords are not given. Maybe it's not given. Because this is giving the console and all
Yeah,
that.
because this is substance.
Okay, no problem.
We can go with LS.
Okay, just forget about that. Okay.
क्या हुआ?
No, just forget
See,
about
you
this.
have, no, it's underscore, it's not dash, okay? New, it's underscore.
We should be able to
So, such file or
From
There is
CD we have to
CD,
write.
after that, it's wrong.
So look here. I mean, because this is not a, I mean, kind of you are using it from the Windows, there might be a problem. So in Linux machine, whenever you give WHO, it will show you the different users when it is created. Now you put WHO in this kind of forward bracket. You see users.txt means you are putting all these things in this particular file. Okay. Now you are telling sort. Sort means you are sorting this user.txt. Okay. So these are see two different comments, but this is a use of this pipe. Okay. So whenever you see the pipe, that means it is telling first do the who command, then do the sort. Okay. And it can do simultaneously like this thing. Okay, it is sorted by this thing from previous one. Okay. And you can cut D or F3 means the third column you can just omit, just put the this column only 1, 2, 3. So this thing only left here. Okay. So this little bit of program you can do here itself. You can try it later on. I give you all these things. But now try to identify these wildcards. Okay. Wildcards are very important here. Like in search we used to do, like I have a file name say start1.txt. Okay. Now I forgot that name. I want to find out all the file names starting with start. Then what you can do, start, then start.txt. Then it will give you in your Windows system all the files which is starting with the start. So here star means also in Linux also matches any number and kind of character. Okay, like usable with most Unix program like start.fasta means it will consider any .fasta file in any folder. Okay, suppose there are thousands of FASTA files are there. You want to consider all of them in together. So you can use start.fasta. It will consider all the .fasta files. Like when used as a parameter value to a program, the OS finds all the files matching the pattern. Like soil start.fasta. So it will find soil1.fasta, soil2.fasta, soil3.fasta, like this way. Can be used multiple times. Okay, like r1.fast, fa1.zz or something like that. Okay, so star means anything, and question mark means matches a single character. Star means any number of characters. Question mark is any single character. Here it will be only soil 1, 2, 3, 4, something like that. Okay, so these are wildcards. So project management and naming things, that is very important in this programming. Store things in logical design location and stick to it. Okay, please plan it properly. Okay, before and where you are putting your data, and logically put the data like raw data, then after filtering in filtered data, okay, then in processed data, in something like that. Name rules, the name should be machine readable. Don't use any space. Okay, don't use any space for any file name or any folder name. Okay, machine cannot read it. Okay, so name rules, it should be machine readable as well as human readable, and play well with default ordering. Suppose you now you are doing these things. If you come back after 1 month, you will forget what you have done. Okay, so in order to rectify that, try to give file names in such a way that later on you can identify those things pretty well. Okay, like avoid space, special meaning in Unix. So avoid, like this is a very bad naming. Sum space dna.fastq.dz, fig space 2.png. This is very bad naming. Don't do it. You can do this one, hns 214 r1.fastq. Okay, fig 2 histogram amg counts svg. So try to give full names. This is a figure 2 which is depicting a histogram from a particular study and is from the count. So this is a very good naming. Okay, common. Okay, so in a folder, so if you type ls, it will show all the files and folders present in that particular file. cd means change directory. If you want to change directory from one file to another file, you can use the cd command. pwd will give you a particular location. I think I have all these things. mkdir is the making directory. cp is the copy a file. md move a file. remove file. So grep is a very powerful command. Okay, it's from a file or anything you can grab a particular thing. Change mode. Then another important file is gunzip or unzip to, and wget. We use wget in order to download from internet. Okay, just type wget and give the https the whole link. It will be downloaded. And go to the folder and type this wget and the link, it will download all the things. Okay. So look here. So here is a system like root, then bean, etc. Users, temp, user, max. There is a mean another folder. Work is a folder under max. Work and play is a folder under max. Now, if you use first ls minus l, what will you see? Type ls first here.
ls minus I.
Everyone knows their directory, na?
Sir, space minus
Yeah, space is very important.
Space is important.
minus
Space is very important.
Dollar.
It will show all the read write permissions.
Exactly. So only ls will show the files only. But with l command it will give the permissions whether the files is given read write permissions or not. So that you can try. Now from here I am doing cd then space users max work. So where I am going? From here I am going here to here to here. I am now entering in this particular folder. Look, cd, then I am using users, then max, then work. So from here I am entering into that particular directory. Okay, you can also write these things cd dot 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 dot. This is absolute, this is relative path. Okay, so now I am in the work folder. And whenever you write pwd, type pwd, write pwd here, just pwd, type pwd. What it is giving?
Pass.
Path. So if you don't sure about where you are in Linux system, if you type pwd, it will give your path. So it will can recognize you. Okay. Like here, if you type pwd, it will show like this: users max work. So you are here actually in work folder, under the max folder, under the user folder. Okay. So now I am doing in this folder what I am doing? I am doing users max play 1 mkdir. I am creating a directory in named it play 1 under max folder under user. So under max a new folder will be created that we call mkdir. So if I am here and if I just type mkdir play 1, then play 1 will be created in between 1. Okay, but here see I have given the full path where to create the folder. It's under, it should be under the max. Okay, but if you are here and just type mkdir play 1, then the play 1 will be created here. Clear? Very good. So now this is another command cp. You are copying play 1 where going to users mean. So you are telling that play 1 folder you want to copy here. What will happen? The folder, this thing will go to here. It will be copied to here.
Should we give space in between?
Yes, space is very important. Space is very important. Here is space, here is space, here is space. Okay, it's very important. So you are copying this folder and you are putting it. You are going to users and you are putting it here. Okay. So unique comments to know: mv is renaming file. You can rename file. mv rm you can delete files. Change mode you can share files for the sharing options. Change mode plus x. Some files like if you download the usearch and other files, you have to give this permission. Okay, making it executable. So usearch file is you can download it, but it is not in executable mode. Okay. So you download it and if you give this command, then it will be in a exe mode. Then it only can run. Okay. Grep I already told you. Suppose you have this cat, it will show you the whole file content. So if you want to see the whole file content, just cat and file name. It will show the whole file content. So in this file, don't try it with the big files like GB, MB level file. Don't try it. Don't do cat. Then system will be in stress. Okay. Head, instead use the head. Okay. So here it's a small file. I have used cat and you can see the file you can see. Now what I am doing? I am telling grep gtt from rrna.fa. I am telling find out gtt from this. Now it will show where it will find the gtt. That's a nice thing, grep. Okay. Suppose you have multi-FASTA file. This is a single FASTA. Suppose you have multi-FASTA file like this. So now you are telling just grep, this sign tells you that at the beginning of line. This caret sign is telling you at the beginning. So you are telling to find greater than sign at the beginning of the line. And it will show, look, these three lines they have just extracted and it will show. So in any faster format you can, this way you can identify the header files. But suppose you want to count it, what you will do? You will first grep this thing, then you use the pi and then WC minus A. Means how many times this greater sign is appearing, count them. It will only be 3. So for any FASTQ files, if you do this with @ sign, you can count how many FASTQ files are there. Clear? Okay. So other things are there, curl installed by the default on most system. wget is very important. Okay, it can handle recursive downloads, folder contents and folders below. So like this. Okay, wget, then the FTP site link, and it will download this content from this FTP site. Okay, and download and activate usearch. Usearch is another very important. I told you. Okay. From the usearch you can create the OTU files. So like you are creating a bin_temp directory. Okay. Now you are entering into the directory. Okay. Now you are doing wget. Okay. Now you are unzipping that thing. Now you are moving this, renaming this thing such as usearch. Then you are giving the permission. It's executable. You can do it right now if you have time.
Yeah, do it. Type this. You first make the directory.
Yeah.
mkdir, whatever
Then you
name
have
you want.
some idea how
Yeah.
to do all these things.
You can give the same name, bin_temp. You can do it, yeah. You can do it. Yeah, you can do it. So you are in the directory now, you can do it.
But this one is not.
You type this one now.
bin_temp na. Then you can go for cd
What you are doing, Balaka? You type this, that one, na?
What is this?
No, that. Don't, W can why you did that one, na?
yes
To directory.
wget.
The system information file. No, you are not in directory. See, if you are in directory, it should come like this, okay?
Disable.
It's same line, right?
See,
Yes.
after this wget we are getting like this.
wget is already there, na sir? After wget, we have to give.
The path.
We have to give the path, the link, the HTTP link, whatever you want to download, that thing we have to give. wget is a like command to get the download that part. So now you have make the directory. See, mkdir, right? You have mkdir means make directory. So you are giving the folder name is bin_temp, okay? So what you have to do? You are in the directory like cd mnt c users. Then you are in the folder like new set 1. So you have to go there. You have to minimize this one, and then you have to go in your desktop new set 1. And there you will see one folder has been created. That name is bin_temp.
Please check. First make directory bin_temp.
The bin_temp, you can give any name according to your choose. That is the folder name. Like normally we are doing, that is only folder name.
After that you please check. Are you getting that bin_temp file is there? Yes.
Oh, Linux.
bin_temp is there? Hey, all you got the bin_temp folder is there?
तुम्हारा कितने दो?
Folder is there? bin_temp.
Just download it.
Okay. So next
Got it, it's done.
all got it? bin_temp.
Not found.
Not found.
You go there, the new folder one. No, आपको गलत हुआ देखो bean underscore tmp. There should not be any space. You see the command.
Command and type it here.
Space
This two, suppose how they
They have already downloaded that part. Okay. So W get, after that you have to give one space. Then you give the path.
उसमें स्पेस नहीं है TMP में
After CD bin you have done, and after that dollar W get, then have one space, then you type all these things. HTTP, www,
Go to that file na, this lady.
wait, which side?
Slash between D and slash between D and slash.
not found I saw it.
Then 0.667.
Underscore
Space, second one. W get space,
W get space. This
and then HTTP,
6 Linux 32
then this W get drive.
32 dot.
Wait, alam mo ito.
That's it.
Not,
32, na?
Why?
I think there should be less regulated timing.
Like we are creating one folder and we are saving this link video in that folder.
Error is there, error is there.
Sir, after giving
You make the
this W given,
Yeah.
it's not getting
So you please try the link itself in the
any name of folder you want.
Go to the Google,
go to the
Google and they get the link from there.
Yeah, so whether the link is still active or not, that we need to check. Okay. So you can do this.
Yeah, it's working, it's working.
it is
working.
They got already.
Then it's fine. That's fine.
Type the command carefully. There should not be.
So yes,
32 only we have to get.
Yeah. You get 64.
64, you have to buy 64. You have to pay more for the 64, like $1,000 or something like that. Only 32 is free. 64 is not free.
then we can go for the unzip also.
Yes.
Okay,
Then
next.
you go for the unzip.
Unzip.
So it will be unzip.
Unzip. You search.
32 days.
Installing users, you can go directly in Google. Let's go up.
The file should be there or it's actually it's not there. Is it just
Down.
it's not there? Which means that wait.
So that's the thing I was talking, you are using now. So this is the thing you are using now. Okay. So package management, containers, virtual machines. So there is kind of one important thing is your analysis or application. You can do the Conda environment. This is the environment, you create the environment where all your supporting kind like this kind 2. You can install this kind 2 in Conda environment. Okay. So that it will be a stable thing. Then Docker is more stable. Okay. So in Docker everything will be set like how to install, where to install and other all these things. Everything will be preset. And the virtual machines, these are kind of you can use, but although the virtual machines are not pretty good, I mean it's a bit slow, and installing these virtual machines is a tedious process. So mostly we use the, once you have the Linux, we use the Anaconda, okay, or Conda environment, we call it powered by Continuum Analytics. So this is a kind of environment, means all the supporting kind of programs with a pipeline will be given to you at a one single go, okay. So now we will use this Conda environment in order to install the KIME2, okay, KIME2 package. I think it's already installed, yes?
Yes.
Okay. So now go to there. Here, I think. Okay. Now please show it how to install. Then I'll give the.
Can you show me
Okay.
how?
Now show it. So first wget means you are downloading the Miniconda. Okay. This is the link for Miniconda download. Then you create just bash this Miniconda. Then you are creating this Conda environment for KIME2. Okay, it is activating. So then you are just first activate means you are creating first the Conda, this KIME2 environment. Then again in the environment you are downloading the KIME2. Okay. Then you are creating Conda environment, create KIME2. Then you are activating here. I mean, you cannot activate here. Before you have to create, then you can only activate. Then you are entering into the desktop new set, and I think that's all. Okay. So then you are done with it. Then I have some example sequence files with which we will work. Okay. So we have I have some FASTQ files and metadata files. So we'll start with that. Okay.
Give them in a, I think we have to give them in pendrive.
Yeah, I think.
Pen drive चाहिए.
To copy all this text.
Okay. So these are the, you can see here. So these are the steps we do. First, look here. First, we'll import the sequence data to KIME. So KIME is kind of a KIME2 is kind of a platform. So first, what we have to do, we have to import the first Q files to the KIME. Okay. So these are the kind of codes we comment we write, we use, then we visualize the data and how it looks like. Okay. So then the next part is denoising the sequences, selecting the sequence variants and feature table construction. So this command itself will denoise the data, then it will select the sequence variant and it will create the feature table. Once the feature table is generated, we will generate feature table and feature data summaries. By summaries we can see how many features are there. Then filter the feature table like low quality total frequency based filtering, like what we did in case of microbiome analyst. That we'll do it here. Then again we'll filter the features with very low abundance. Okay, so very low abundance features with OTU files will skirt. Then we'll use the green jeans version, this 13_8 version. So for the taxonomy assignment, and for this we'll use this name based classifier artifact. So this is a predefined trained data set. Okay. So this classifier is already there. Based upon this classifier is created based upon this green jeans database. So we have these things. So we will use this thing. So where the sequences have been trimmed to only include 250 bases from V4 hypervariable region of the 16S and pre-clustered at 99 sequence identity. Okay. So we will obtain this from KIME2 site with the command. And you can just, this is the link where you can get the classifiers. Then we'll use the classifier, a sklearn classifier for performing the taxonomic assignment. Then we'll create the taxonomic table. So now we have the taxonomic assignment feature table with us. Now we'll visualize the results. Okay. So first we will remove the unwanted taxa from the table and the sequences. We will then filter the tables, and then we will remove the archaea because we are interested only in the bacteria. Then filter the eukaryotes, and then filter the sequence with mitochondria or chloroplast. These are kind of impurities present in the sequence. We need to remove them. After removing them, we will visualize the taxonomic classification. We will create the bar plot, what we did with the microbiome analyst. Now we will build the phylogenetic tree for phylogenetic diversity analysis. For PD diversity or unified method, we have to build the phylogenetic tree. So this is the method by which we create the phylogenetic tree. Then we will identify the alpha and beta diversity analysis using kind 2. We can visualize the result, then generate the alpha and beta diversity matrices. Then we will do the alpha rarefaction, and then with the rarefaction we will do the faith PD, Shannon index, and we will visualize the result. Then in the beta diversity we will do the weighted unifact, okay, what we just studied. And last we will create the emperor plot, and we will visualize these things. So all these things you can do it in kind 2. So that you please first learn. And yesterday I have shown you, if you just assign the taxonomy and the feature table, that you can convert into the biome format, and along with metadata you can also use first you find, I don't know, accordingly we need to extract the classifier.
environment create करने के लिए स्टेटमेंट चलाने के लिए एनाकोंडा की रिसोर्स फ्री डाउनलोड किए
Yeah, because these are for metadata for development to see. And here we only have 36, remains 18. Okay. Here we are 18. Do you have the previous data? Because
Yes, it's there. Here in new set one. I think other thing we delete it. The parent sequence is there. And this is our classifier. And the data is there. It's there. New set one. Sample data.
So look, we'll work with these actually samples. So we have in total 14 samples, okay, here. So some are NO, and some are HHH. So these NO samples, they are infection samples, and these H samples, they are healthy samples, okay. So we'll first look how the microbiome is different in these infection samples versus these healthy samples.
So we can show the other part, how what you have to copy and keep it in
Yeah,
another
and this is the kind of FASTQ files. Look, for a particular sample, two FASTQ files are there, R1, R2. So these are forward read, these are reverse read. And these are the sequences actually we got from the Illumina. Okay. So Illumina reads are there, R1 and R2.
Okay.
Now you have given all the files.
Yes, all are there. So in your desktop, go to the desktop.
Go to the desktop.
Yes, sir.
Yes, go to the desktop. Gone.
Yeah. Everyone in the desktop.
Yes.
Yes.
In desktop.
And then see in the desktop we will get one folder called new_set1.
New_set1 is there?
Yes.
Click that one.
Yeah, open that. And then copy the paired and sequences.
Copy.
Yeah,
The
just
folder.
select that. Wait, select
First.
that paired and sequences.
1 minute, 1 minute.
Everyone selected?
Yes, sir.
Selected, 1 minute, yes. Selected pad and sequence.
Folder paired and sequences.
1 minute, pad and. Pad and. Right, next.
Next is GG, that classifier, GG-13-899, that classifier file.
GG.
Dot Q is a
Yeah, GG13.
That QZA file.
1 minute, 1 minute. GG13.
This file and this file and this here.
Yes. GG13. Then
Done.
And sample metadata.
And then down you will see one sample metadata.tsv.
sample metadata dot
TSV.
TSV.
Yeah, that select that one.
TSV. Sample metadata dot TSV. Selected.
Then copy. Copy that.
All copy. Right
Copy
click.
and make one folder in the desktop itself.
Copy and again go to desktop. Then make one folder.
Like time2 run, time2_run.
Time2_
QIIME
QIMQ_.
run.
kind QIIME time_run RUN.
Yeah, try to write in small letter, okay?
small letter.
Kind.
So now show the first part.
And then paste it there. All the file, paste it there.
Yes, you have already opened that folder. Go there.
I have
and
already
paste.
copied.
Now look into that folder.
Paste. Pasted.
Now first look into that folder.
All folder you have. You please look into.
There was total 3 files, na?
Yes.
3 files, na? Paired
Yeah,
and
one
sample
thing you
metadata.
look.
Remove this.
What it is there. Just you see.
Yes.
In that
Yes.
folder there should be three files, okay? One is
Yeah.
all the sequences, one is the sample metadata information, the another is GG, that classifier file.
Okay.
Correct?
So now open that folder, paired end sequences.
Pairs and the sequences.
Yes.
Now what you can see, look, 28 different files.
Yes.
Okay, now sidewise please open in Excel. Please open in Excel that sample metadata.tsv. It's open.
Yeah, open one.
Yeah.
Excel. Open.
Blank.
Blank. Yeah.
File open karo. File open.
File open.
The sample metadata file where it is there in desktop.
No, you have to browse.
Browse, go to the browse. Down.
Where you are giving desktop, no?
And then go to the desktop and then your time to run. Open the TSV file.
Open.
All file you go do it, all
No,
files.
just select
Do it
all files.
Yeah.
Here only the Excel files are shown. Add down. No. Go into that folder again. At that end. Look, it's all files. Yeah. Make it all files. Go up. You are in that folder. Go to the run folder. Now you see the sample metadata. Open the sample metadata. Yes. Open.
Not this one.
Then delimited.
Sir,
Yes.
this one, huh?
Next. then next,
Sir,
then
this one, na?
finish. Yes. So now you can
Infected or
see
healthy?
the name of this sample ID. You have given the sample ID versus this name, the sample name.
Yeah, I think
Just check here, okay?
Excel sheet.
Can you found any similarity? These sample names and the sample ID, can you find? Look, all of them got a, like it is starting from 801 underscore S1, 801 underscore S2. The later part is kind of common to all, okay?
Yes.
But first part, that is actually linked with the sample metadata.
In desktop, no?
Okay,
Desktop.
like 801, 801, N01, N01.
Nothing open.
Okay, so that is actually matching. So you have to create metadata in this way. You don't have to give the full name of the file. Just you have to name the, the unique part, and the other common part, it will be taken
Keep that one in
by
desktop.
itself only.
Copy
Okay.
and keep in desktop. Okay. This one, na?
For you it is not a PDF. It's in first.
Okay. So, and look at the metadata file carefully. Its barcode sequence is given. So you may use the barcode sequence or you may not. Linker primer it is given, but if this sequence is already, we have removed the barcode sequence and linker primer, so it doesn't matter. But look at the stages. Okay, the two different stages are there. Some samples, 7 samples from the infection stages and 7 samples from the healthy individuals. Okay, we'll compare between these two groups, one with the infection group with the healthy group. Okay, we want to see in infection group how the microbial composition actually varying, how the diversity indices is varying in the infection group compared to the healthy group. Okay.
Hey, look at that here. Healthy and infection, correct?
And
So
you
slowly
must change
we are going
your
to
folder name.
compare.
It is not okay.
Here the
All are having a,
everything is open in PDF.
yes.
How you open this in PDF?
No, it's actually
Or you did anything?
very copy into this one.
Ah,
Can you go to this new section, new section also
copy then come first. All files will come.
Next thing, next.
period.
Next.
Yeah, एक दिन में नहीं
Okay, all the, you are having a, right.
Yes.
All finished?
No, for heart.
Oh, here, okay.
This name is changed. Because it's kept, no? Try to keep in.
Just F2. Just click on it and then F2.
Just change in small letter, no? QII.
Not QU.
QII.
QIIME.
2.
Underscore run.
Underscore.
Run.
Oh, you have to copy them. Oh, hard sir, all
What?
the files are copied in Adobe
PDF, sir. What of
PDF.
No, not in PDF. Just look at the extension. It is first view, but somehow somebody opened.
It's like they unzip
It will
them.
work. It will work. It will work. It will definitely work.
They unzip them.
No, it will. QIIME.
QIIM. Because give the same name. Q small letter QIIM.
Sir, like whenever we are getting after sequencing, the company will be providing these pre-data.
Because your file is open.
Something is open there. I think Excel is
Excel
open.
is open, no? Yes.
Open it.
Now is right.
Okay. Now just show the next gene.
Okay, you find out what is kind.
Kind 2.
What is kind?
Yeah, kind next generation microbiome.
Yeah, that's fine. Now start the next thing.
What is kind?
Go to here. Okay, now this first thing, the
इसका behind.
now all of you please look at here. Okay. Now the first things we will be doing.
So first, now we have this folder, right? We all copied everything for the whatever prerequisite thing is there. Everything is there in our folder now. We have paired our parent sequence. We have that classifier file. You know what is classifier file?
No.
Sir already discussed that time. The reference file.
Reference file.
Yes.
That green gene, then Silva, the database with whom we are going to align our sequence. The reference gene. Okay? This is green gene you have already downloaded. Even you can download by already command are there, you can download it. So we are keeping here. Right? And the sample metadata file. Here all the information.
So this above file has come from Illumina.
Yes, these
First
sequences are from Illumina data.
Okay. And these two?
This one you have to download according to your need, okay? If you are targeting V3, V4, you will have different classifier file. If you are having only V4, then you will have different classifier file. According to your need, you have to download it. You will get in the kind 2 forum itself. Yeah, you will get it. It's in internet only. And this one you have to prepare. So sir again and again emphasize this part. This is very important part. So according to your hypothesis, like work design, you have to design your sample metadata. Like what parameters you are going to compare. So this according to your need, you have to modify this sample metadata file. So it should be in TSV format. Okay? So now everything we have, now what we have to do, we have to import that sequence. Yeah.
This file is there.
Oh, they don't have that file. This file. You have to
It
copy
will be
this.
helpful if you have.
Because they can copy. Yeah.
Yeah, make it a PDF and send it via WhatsApp or something.
Yeah, that's good. I will send in the group.
No, otherwise very difficult. You have to copy all the things. Otherwise they can copy and paste, na?
Yeah. Just
Copy
copy
in our system.
this file.
Pen drive.
Pen drive there.
Where is it? Your comment?
It's not there. It's this one. I always tell to don't copy and paste. I always tell to write it down by yourself.
Yes.
Yes, sir, I
Otherwise,
agree.
you cannot understand how to write it in the
Yes,
command
yes,
prompt. Okay, don't copy and paste.
Okay.
Yes.
Then you cannot learn how to write it in the command prompt. Because writing command prompt is a different.
Because you have to be very careful with space, everything.
Exactly. So don't copy paste. Don't copy paste.
This third, according to needs, is we have to prepare.
Exactly. I told it metadata about the metadata. So we need the metadata for each and every sample. All the details will be given. Okay. Like where did you isolate the sample? Where there is a healthy sample, or disease sample, or something like that. So as per your needs, you can create the metadata. Here in this project, the objective is to see whether there is a microbiome difference between the healthy group and the infection group. Okay. So we just want to see whether in infection group microbiota changes from the healthy group or not. Okay. So that's our objective. So that's why we took 7 samples, each from the healthy group, 7 samples from the healthy group and 7 samples from the infection group.
As per your experimental design, you will change.
You will change.
You can keep separate so many things there, column wise.
Suppose if you are having 4 groups, 5 groups, 10 groups, accordingly you have to make change.
You have to make
Like age, like gender, you can add the age, gender.
They will form.
Yeah.
The index, these are the barcode, no?
No, barcode and this linker primary.
Exactly. This will give you by the company.
By the company.
Company, you have to ask this
You have to
first.
ask, otherwise they will not give. That is why this barcode and linker you are asking, they will give. Then accordingly you have to prepare.
When you can open the sequence, you will see the barcode and linker primer part. It will be there in the sequence. You see N01. You open the sequence and you will see the barcode and the barcode plus linker part.
All first open one file.
Wait, I will put here.
It will take too much time.
Yeah, we have
For the
to
size.
It's not that necessary.
Then you can.
Yeah.
Yeah, this one, huh? This one. This one
Just
ask
wait.
you.
I will show here. Wait.
Yeah.
Just wait.
18 MB.
Yeah, it will. Just wait, huh?
It will take some time.
Could it sit in a bit?
This system is very slow.
No, whatever it will not open.
Notepad.
Open.
Not pen.
Edit pad or something like that.
You open one file. Are you in a position to
You
open?
can open it in the Linux
Go to
system
run
by the cat
paired
command.
sequences.
Yeah,
Open one
go to
thing.
Linux and use the cat command.
Right click. Right click.
System option.
It will take some
You
big
need to
file.
right click and open.
Choose which.
Actually,
It's not
this
needed
gzip.
anymore.
Yeah, which file will it
Notepad it will not open.
Yeah, go ls, ls. Yeah, then go cd, paired in sequences.
CAT.
cd,
Okay.
paired in, then tab. Use tab, just, no. Then space, then right P.
What do you want it?
Use tab.
Okay.
It will give you,
Notepad is open. Then choose answer as
then enter.
select
Now
word
ls
file.
section.
They are also
Now
facing.
cat in your.
They are also not opening.
This one I need.
801. Right. 801. Capital H. Total is
Not
01.
opening.
Then tab. Use tab. 1 tab. Yes. So you need to unzip.
This.
See, it will already take. Right here. So it's in the zip condition, so unzip that thing, okay? Now ls, then unzip one of the first file.
You will
Use
be
it.
doing that simulation.
Ganji.
Yeah. Ganji space
Space.
801 capital.
Okay.
R1
Space.
tab.
R1 tab. Then cat.
Ls. Let me see. Why you are using tab now? Yeah, it's there. Now do the cat space. Yeah, it's there. 801 tab. One tab. Yeah. Enter. Now see. Here.
Beginning.
Okay. So this is the first file. First Q file you see. Okay. This is the header, and this is the sequence, and this is the last sign. Then this is the quality value. Okay. So what do you want to see here?
All code and index file.
Primer.
No, those are already trimmed here.
Linker primer.
Those are already trimmed.
Yeah, but untrimmed data will get.
Yeah,
In my
in
data I have seen.
in the
Yeah, the raw
untrimmed
data,
data,
yeah.
In untrimmed data, you can see here, actually at the beginning. or
Yeah, at the beginning.
At the beginning, you can see the
After
bar.
the,
Yes.
just after the header.
Yeah, just at the header. You
What?
can see that.
I will show, okay, in my data I can show that.
But
Because
here,
it's
no
already
need-
trimmed.
yeah.
It's already
So the company will provide this thing, okay? So now we need to remove that thing, okay? Now start that thing.
Everyone copied that command?
No?
All command is there.
No, wait.
He is busy with this video.
Videography.
So no, he's trying to put it in your head, not in video.
Professor Basim also told the same thing.
Please try to put it on your head, not in the video. It will not
So much thing we can't put
It
it.
You will
not
get the
at
video.
all- It will not at all be helpful for you.
Just wait.
It's totally wasting of your time, nothing else.
Yeah, you should pass now. You are not copying, but you are keeping the pen drive with you. You are copying.
मेरा पेन ड्राइव है
Pass.
नहीं मेरा पेन ड्राइव
Okay,
है
रुकिए.
उसमें नहीं है उसमें नहीं है
It's not there.
it's not there
Not
it's not there
copy it.
अरे,
Copy it. Copy on. Copy it. Copy in that pen drive. Just wait. Copy it. Isn't it? Sir. 85. Sonia, and you copy it.
Now open that file. Open that Word file.
Open your word file.
So different steps are given here with all the comments. Okay, now you can see which are comments, which are parameters. Yes. So previously we discussed about all these things like kind tools import. Okay. So there are flags we are using like the type, dash type. That is a what type of sequence we'll be using. It's a sample data. It's paired end sequences with quality we are mentioning. Input path, paired end sequences. Input format. Okay. Output path, the demultiplex paired end dot output will be that thing. Okay. And input path we are mentioning paired end sequences folder there. Okay. So go to that folder using this command prompt. Now go to your Ubuntu.
Go to Ubuntu.
Yes. Now go to the desktop where you have put your folder.
Yeah.
Just go to there.
One by one you type it.
Should be open.
Yeah.
So new one we have to open.
Yeah. You check your that word file na.
Just check it ls.
Yes.
So you are in the time to run. Very good. Go to the time to run.
So the folder what you have created, just go last one na search. Whatever it is created, just go
So
to the
do
folder.
one thing. Type ls here. Type ls.
Type ls after going to the
Now
directory.
you can see where you are.
आपने
You are
कॉपी
in
किया है?
users. Okay.
कहाँ पे कॉपी
So
किया है?
you have to come out of that folder. Then you write cd dot
टाइम टू रन ना तो आपको इसमें जाना पड़ेगा.
space dot dot space dot dot cd then space then dot dot. No. cd
There's new one only.
yes then space enter. Okay, now you are one set above. Okay, then again do cd dot dot cd space. Now see you are in the desktop. Okay, now you have to enter where cd time to run. Right, cd space Q, then you tap. Now look, you are to change PWD. Just enter into the time to run.
Got it.
Just wait there.
All are there, right?
Yeah.
Once again.
Yes, yes. Hey, please check here. Yeah. She kind to underscore run.
This is the next campus.
Yes, let's go.
This people done several times?
No, I think
Okay, do it. No, it's not correct. How many times have done? Huh?
Kind to underscore run.
Now where are you now?
Where is the
Type
type?
PWD. Yeah, PWD. Enter. It will tell you are in home underscore GG. Okay. So you don't know where you are.
Write it
Yes.
from your mobile.
So now do ls.
Yes.
Enter. So look, you are in anaconda command. You are in a very bad position. Okay. So go to do cd space dot dot. Space dot dot. So you are going one step above. Now enter. And again give ls.
Yes.
You are cheating.
Now see you are where are you? Okay. Do one thing. Go to your folder. That time to run folder. Yeah. Time to run. Go to time to run.
It's showing you.
Kind to run. Click here.
What are they listing?
Why you have closed the? Okay. Just click above. Yes. Click above. Yes. Now copy it. Okay. Now go there.
Show the path.
Why mega? Now cd d space. Now paste. Now enter. So something happened here. This is GG. Why GG desktop?
You did just now.
No. So you are in also the GG desktop, yes?
Yeah. All are doing the thing only.
Yeah, but why it's not coming here?
So there's two files.
Huh?
You separate.
Okay, now again enter into the GG. Like write CD GG. CD space GG. Enter. Okay, now again do this thing. Yeah, enter.
All the files inside the folder.
It should come here. No.
It's not correct. You didn't do mount. Mount switching.
Yes, sir, it's backslash, not forward slash.
Before you have to change this thing.
Forward slash. It's backslash. I will do it.
Like this. You have to do this thing. No, they can do it. Okay, change these slashes. Yes. No.
All the
Before Lenovo.
all the
Go to
backward
the,
slash.
yes. Here. Now go up. Go another step. Yeah. Now delete.
Backspace. Now backspace.
Now delete.
Oh, delete.
Delete.
USER S
Here.
S.
Users.
After LS, you should do PWD again. No, I'm asking you. You should do PWD again.
Now do it. Again?
No, I was asking you,
Now
like,
do it.
Now write CD first.
I'm asking because
First
I
write
don't
CD
know.
space. Now do it by yourself.
Now do it by yourself.
Delete, not backspace.
List.
Yes, that's fine. Yes, and delete. Yes. Now enter. Huh?
Because I will do it, sir.
What happened?
No, sir, because I understand.
So at first you used to
Oh.
CDMNT, sir.
Yeah.
I will.
Because here you have to do the bounds.
Yes, yes, sir. Because this is.
good here.
Because this is not
Direct
real,
system.
yeah, real system. So you have to mount first. Okay. So like previously for the pen drives and these things you have to mount first. Okay. So now write PWD. Write PWD. You can see your path. Okay. You see. Enter. Look, this is your path. You are there in the kind2_run. Now show it. Show it. So all of you are in the kind2_run folder. Okay. Where you have paired sequences folder, one metadata file and one classifier file. Very good. We are done with this.
Yeah, now so what we have downloaded for this analysis, we have downloaded Anaconda, right? So we have to create the Conda environment before going to the analysis. So how?
Container is already created or not?
No, so we have to activate again and again.
Just no, you can activate.
Yeah, we have to activate that. Yeah. So we have to activate that Conda environment.
Because he already created the Conda environment. So as I told you, this is an environment where the kind2 program is properly installed. Okay, with all its supporting programs. Okay, so now she has already created it. Now she will activate that particular environment. Okay, that means you are activating kind2. Activating means you are just calling that program so that you can do work now. Okay.
So for that you give the command Conda space activate.
Before activating, how we should create? Can you give us just how?
See, it's there in the command is there.
Look, this is the
Command.
yeah, in H3 you can see it.
Yeah, you can write Conda space activate space kind2, the version of the kind2
No,
you give.
she is asking how to create the Conda environment.
Later I will
So
tell.
that actually I have shown to you. I mean in the Linux that slide I have shown to you how to create and all. So that is already there. Don't worry. Okay.
We will do it later.
You can do it. But now just learn this thing. Okay.
Honda space activate space kind2 then dash 2022.8 enter.
Yeah, after that, after writing this, enter.
Enter. Enter.
No sir, let them write. No, sir.
Active.
Done. Everyone done? Can 8.8.
Just 1 minute. It is written there. Look, Conda n means environment. Create, then minus n kind2, then file. So that's the command. It is already there. In history, we will later on get it. Okay. Just note it down, and later on you can get it. All of you activate it.
Done?
And see, you can see a big picture of anaconda. Yeah, you can see a big picture of anaconda snake. So then you can think the program is running properly. Otherwise, the program is not running properly.
So
Have you got the snake?
No sir, we are not using here. This one we will not take. This is not real one.
What is there? Yeah, you don't know. You go to Champaya. What is there? Dampa, you go there. Yes. You are there in Israel.
So done na?
So now look. So now beforehand it was base.
Yes.
Look, base root. Now look, it's under kind2 2022.8. So you have activated kind to, and you have entered into the program. Now you are ready to do the analysis. Now look at that first import thing. That word file, yes, here. So first, no, please don't copy.
Hey, don't copy, yes.
Don't
Let's
copy.
write it because see here it is backslash is there.
Ah.
So we have to work with forward
All
slash.
changes.
And then this is in all the commands should be in one line. Okay?
So look here, just carefully read it. So what first we will be doing? First we will consider all the FASTQ files. So it is called importing those FASTQ files into the Conda environment. So we will be importing all the FASTQ files into the Conda environment. So we are calling kind tools import.
Yes.
We are calling import program here command. Now telling what type of sample it is. It's a sample data paired in reads.
With quality score.
Yeah, with quality score is given. Then we are giving the input path. Input path, what is it? Is the folder name.
Yes.
Because you are in kaim_run, kaim2_run folder. Within that there is one folder called paired and sequences. So you are giving paired and sequences as input. Then you are telling that input format, this is cassava 18 single lane per sample direct FMT. Okay, this is kind of company generated thing, don't change it. Okay, this is for Illumina. And now output path, look, now I am creating this demultiplex paired_qza. Qza file is the kind2 specific file. It is a kind2 specific file. That means now all your paired and all the data is now will be present in this dmax paired_qza.
After running this command, you will
Yes.
get, you will see in your folder one more file will generate because now you have 3 files, right? You will see one more file will be generated, that is output. This will be dmax-paired-n.qza file. So now run this command.
So next we will work with this file only. We don't need any more the sequence.
And this slash.
No slash, sir. With
No
single
slash.
line. No slash.
Oh.
See.
No, show me.
From where?
See what she's doing. Yeah.
Now, see, you should run the command just like this. Okay?
So all slash you remove it.
Yeah.
You go to your Word file.
Then what
Type.
type, that is the best option, otherwise
You will make mistakes and
that is the you can learn.
yeah. Otherwise, if you just copy and paste, it will be done.
No, first do it in Word file.
Okay, sir.
Because correct. Gap everything. Then you yes. Yeah.
So for the visualization, you please open that kind of visualization. So that will be easier. Okay. Don't try this visualization.
Let them copy that.
Yeah. First, let
Just
me do-
click on photo na in your mobile, that will be very easy. Otherwise you are seeing just like this, you may make mistake.
Yeah, this is a nice way. You just opened it in a two window and type. This is a nice way.
Yes.
Look, you are doing
Minimize
mistake.
this.
Input then one. Yes.
Minimize this.
Mobile is so handy. People even cannot read from the screen. Okay, it's only from the mobile.
Just minimize the one and then that is maximize.
Yes.
Do it, yeah. Yes. Minimize for
You
you.
can do very well from there.
Space, right?
Yeah.
Just
She can do mistake, okay, please look from there. And if she can do, I mean, she has kind of wrong entry, then you will also have the wrong. Yes, that's the best way. I mean, minimize that thing. Okay, not like this. Okay, now here, okay, now we can see without
There you have to cut that
Yes.
number you can do. Suppose here you want around the
We have to type from here, no? We have to type from here.
Give that command, no?
I'll focus on the system, okay?
so like that.
Done?
From here.
Yes, done. Now the open to mobile.
Here you cut the where you want that exact name you give.
You coming time now? Just to stop this one, okay? You coming time just to stop this one. You are coming time. Okay. After run you are coming now.
Okay,
See,
okay.
just to stop.
Minus.
First I can design that.
Yeah, before here is that. Here is
Here's space.
Here is space.
Dash.
हाँ
Let's check.
इसमें play करना है ठीक है you with your data you have to play here only one more step is there where you have to card where you have to team and only you have to play according to your data quality this will change
तो वो आउटपुट वाला नहीं करना है ना आउटपुट वाला जो था उसका लास्ट में
20 is done.
इसमें कुछ नहीं करना है अभी इसमें ऐसे दे दो जो है वही दे दो
ये वाला स्टेप नहीं करना है
नहीं करना है up to QZB only no not QZB
Output there is no table. Start this forward step.
Excuse me. I turn on the record. While coming to you turn on. Okay.
How to turn off?
Just press this button.
what it is there try
Red color
typing.
button.
That is from
Okay.
Okay.
So see, your problem is here input demultiplex that here your problem is here, not here.
Maybe something happened.
Input demultiplex, that's 6. Then demarks, here space is there. Just wait. See here after that space is there.
Space will be this side.
Here. It's running.
Yes, it's running like
Mine have showed error.
yeah, even yours.
Then
where?
forward
Go down.
and reverse. This one is forward. F for forward, R for reverse.
Yeah.
This forward means
There
up to
was
forward
a
read
problem
R1
with the
up to
command.
200 right
No,
side.
such
And then
that
23 is beginning.
Option.
First 23 after 23 only one
Say
time.
the
That's
such
your
Such
left
option
side.
is not there.
Okay?
Yeah.
This is for forward and same way it is for reverse.
No, put down. 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 Hold down. Here.
नहीं, it's up to you, okay?
अपने आप डिसाइड कर सकते हैं ना
हाँ, अपने आप डिसाइड करो।
उसके बाद जो-
लेफ्ट में कितना लेना है, राइट में कितना
उसके बाद
लेना
जो
है।
हमको बेस्ट लगे उस
उसी बेसिस से जाओ, क्योंकि ज़्यादा तुम ट्रिम नहीं कर सकते।
ज़्यादा
Otherwise
करेंगे
you will
तो
lose
sequence
your
मैं
sequence.
वही बता रही
Or here.
हाँ, you can 0 or 1
Where you
10.
have to put over?
Yeah.
You can but
Now such options by
Fast B is not good because fast to start the luminite, there is some noise.
Double T is there.
Yeah.
Yeah, it will take so much of time.
I enter it.
On activate it.
That's it.
what is the estimate?
Maybe it's not deleting.
Dina?
Oi, just yellow color should be new one.
Double table, here is there. Table.
Yes, but it's time to okay, I'm so sorry. Just read
Times now. This we have. Okay. This. No. Yeah.
EA subsystem I haven't tried it. Conda activate kind 2-2022.8 na. Conda activate point 2 dash.
Shall we go for TNC?
He has to close this.
So M
Sample metadata file. So you are
Now we are-
feature table with the metadata.
right, yes. So
Then you are changing to user data.
yes, so in the second step, see, kind feature table summarize. So input we are giving table dot uza. In the previous step it was generated, right? And then visualization we are giving visualization output file we are giving table dot uzb. So here we are giving input also the sample metadata file. What is sample metadata now?
That was the and 0 or H1
Yeah, use the proper name actually. Like sample metadata, if it is named sample metadata,
Then it is good.
Yeah.
So with that, by using that sample metadata, we are generating feature table. So what is feature table now?
Like ASB file or OTU.
OTU, sir have told, right? That OTU. So to generate OTU file, we need that table.uza file.
What's the name of the sample? This table is their table dot user. Feature table summary minus I table dot user. Now visualization table dot user. User is there or not? Output is user. Okay. Then aim is sample metadata file. Sample.
And if here metadata one dash is there.
Oh.
No, sample metadata file.
Oh, sample, aim, sample, also there is a space.
Space,
Yeah.
dash.
Dash, aim, sample, dash, metadata, that's fine. That dash is not there.
Yeah.
Now it.
We'll focus on program. I already did mistakes. I already did mistakes. I have done mistakes. Still I am not answering that.
Now table is generated.
Can visualize. Now look at the table dot view.
So this one only you have to give it 5
Visualize.
Yeah, so look over there. The number of samples 15, total features is only 162, total frequency 8,450, minimum frequency, maximum experience per sample. Okay, this is not so important thing.
Then some yeah, that one is that.
Table also
But
we use.
features,
Table is
then frequency, but we go to the interactive one. Yes. So here, like sample wise, the barcode is present in 66 samples. Go to down. Okay, feature count. Look at it. Here you see the importance. So you can see here for 806 sample, there is huge amount of feature count. But for others, you see the feature count is pretty bad. So what happened? We took here 200 was here. Okay. And at that time we lost many sequences. No, now it can go, no problem. But I'm telling that thing. So whenever you see this kind of thing, that means something happened with this thing. Otherwise this is a bad quality data. But look, go down, look for NO2, NO4, it's very bad. Okay, so that's a problem. So it's not good. Go to the feature table. Feature table.
Yes.
This is the frequency, number of cycles. This take you is not good. Your sample is mistake. This is not so good. So that thing we need to change. Increase that time. Increase here. What we do 200 or something. That actually you protect. I think this is the before, na? And we took the classifier for before also. This is your sample. So during marking we lost many. That actually you can rerun. Please check out the
Number of base pairs.
yes, yeah.
Make sure number of the pairs or number of the
just change
sample
the
or number of the
Number of base pairs of each.
one table one specific table, just change the number and change the parameters and see how the things are changed. So you read all these things? Okay. So here you got all the details, I mean the sample wise. So you can see for one sample you get a huge number of features, but for others you will get very small number of features. Okay. If you see here, not an interactive sample here. So here you can see, look for this sample say 03 and 02, you got only 5 and 2 features. Even for 805, 802, you got very bad number of features. But for 806, you got 7,486 features. So something happened, either the sequence quality was not good with other sequences, maybe good with 806, or we used a kind of cutoff of 220 that might be too stringent and it extracted, discarded most of the reads. It might happen, okay. So just proceed with the next step and then we'll see what is happening, okay. Show them the next step. We'll see it later.
The free file is generated, right? So first we used the table.qza. So now here, what do we use? Yeah, rep dash that representative sequence.qza. That file we will use here as the input. The feature table along with the repetitive sequence, representative sequence.
So here we will first do the summarize the table that we do. Metadata file will be there considering. So that we already did table 1. Now we do this left side. Okay, repetitive sequence, representative sequences. We will see. Then we use the filter, the feature table. But before that we need to see why the features are so large.
From the cluster, now we are taking the representative sequence from the feature table.
Go to next step.
So for me, we have changes and we will let us see how this comes. After increasing. Yeah, now we see this one. Yes.
Yeah,
They are started
Oh.
because of system problem. Done, na? This one. Lalbaraka, what happened now?
7,002.
Haven't done. It's not completed. Do it again. See. See, what is the mistake?
That's why this problem is
No, you see, this is barcode wise, okay? So, here, this one.
I am already telling you. As a statistician.
Yes.
7,000 problem. You don't know that one fantastic problem. The problem is a lot of data is there, but others are very poor. So now is it possible to remove that one? I don't
The
know.
whole sequence you have to remove that. That from the main file you have to remove that.
Then again you have to do the same thing. Yes. Then you do it, then 214 Yes.
One or two weeks, what will be the down payment?
Mr.
So those things, please make that part. That is not the
Come down,
solution.
no, see here, very important.
Got the-
This is the real.
We need to got the-
Not good. You have to get a minimum.
Visualize this again. Yeah, it's this day one day.
Fine to use, huh?
No, now बात भी करना आप लोग अभी नहीं रात को लंच डिनर के टाइम पे ना it will happen sir but the so this data that you read is ready to count is very important it will run this will run sir you will see sir the cluster will come different way speed will not the significant will not come
It will
What
take
it will tell?
good quality website, bad price.
Yeah, but
it will run sir
this is statistics. Length.
length of the and figure
That we have mean.
Total number of
Total what? Maybe the number of
that is
different but
you also ask them give me another spelling is not
yes.
so I need 80% no sir before Saturday we have right clearly there
But immediately you do analysis.
yeah sir they will give their whole working score now we have to see that then we have to run it
Yes.
that is the
Immediately you do
police you can see that is the perfect sir
9% is 200, 25% what case?
So now you are getting this kind of result. You are getting the
Some 50%. Different.
don't make it per sample how much money. Don't do it like that.
what it is
No more
tell?
Book a lane. Okay, that is number one. Number two, okay, I don't want it bad. Only you give me the
Present thing, maybe that is the lens.
Only read length of the lane. Right? So you are getting it. Immediately you have to do this analysis.
I'm not sure.
This kind of data is coming, you have to tell very sequence.
He was telling something.
This is the, that's why I called the expert now, because this thing you don't know. Now whatever it is there, you will say medical very
Summary.
So this is the thing you have to do.
You can open this file now.
Then next step repetitive sequence वाला करो
इसमें टेबल भी बनाना होगा
हाँ हाँ इस पे kind of feature का repetitive sequence वाला ना representative sequence
you have to inform. Inform. Then stop. Yes. Then they will.
Okay, now. Okay, now run the next step.
What is the meaning?
No, this what we can get
This is what
Now representative sequence, na? So what's our told?
Cluster.
The cluster form one representative sequence.
This is the OTU.
Now this is OTU is not. This is not OTU. This is representative sequence. Okay. So with this you will use the other analysis. We'll see which step it is using. Yeah. OTU already generated that feature table, na? After denoising the feature table you have done, na?
Okay.
The table not Q that B, that is your OTU table.
Please tell this thing to others.
Yes.
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 Otherwise they don't know.
yeah, it is 2%.
Minimum length is this much of your feature, like that, whatever is there. OTU. Operational taxonomic thinking. So minimum length is
Yeah.
this and
It is
maximum length
2%.
is this.
And this is 5%.
Minimum is there, maximum is there. Okay?
This percent is denoting the nucleotides.
Yeah, the length.
Sequence count.
Length.
Percentage and represents.
Yes.
Percentage, this is percentage.
This 9% is
See, of that length, na, that nucleotides is there, na, so up to that 186, 2% is there out of 100. So maximum is 98% of that length. You understand? You see, minimum length is 180, right? Nucleotide is. Maximum is that. So in between, you will get so many length of sequence, right? Different length sequence you will get. So it is 100%, right? So of that 100%, 186 length is 2%. And then 299 length of sequence, that is 9%. So like that it is there. Understand?
Then so, but here, why is 2 less between 9% and 35%? That's what I said.
Bro, see. You have.
Here,
See,
9%.
your height is 6 feet, okay? Arati's height is 3 feet. My height is how much? 2 feet, suppose.
Yeah.
2 feet, suppose. So here, how many people are there? Arati's height. Suppose me and Arati same height. So we are in 2%.
2%.
Right? Your height who are there? You are alone. That means you are 1%. Understand?
See, this is the
Yeah.
how many percentage is there? Here 162 sequence count is there. 162 is there. Out of that, what is the minimum length? 180. Here 342. Okay, we went 180 and 341. What is the percentage? The 10. So now you see here, out of 98%, 341, 91, 336. Major thing this side is very less is yes, is 2%, this 9%. So that goes okay.
It is having more pounds than
Just have to see
frequencies are
so we have to create the sequence number of samples already number of picture is 162 yeah see 160
so I have to that's also what
Let's go for next one.
Yeah, for next is
kind to see what is the minimum maximum length is we miss this one and the standard deviation is 1 means
What we are going to do now.
268.13 plus or minus 40.1 statistical value 180 to 342 what is the percentage of This sequence is there 186 bibliotext. But is it 341 up by 98. You can just type it and doing maximum. So now you know that you add a most of the thing this volume higher feature table. And it is everything below 50%, then that is why when we start going on, we are fixing per sample what is the cost cost will
This one, sir.
and they are satisfied, otherwise
Just look at here.
Then if it is done, go for the next one. Now this step you have understand, right? Repeat representative sequences.
here in which file OTU is that?
Table.qzb. Oh, oh, that's
Where your OTU is there? OTU when it is there? In which file it is there? No, are you clear about let's say demultiplexing, demux is there? You know everything. So here where your OTU is there? Constant money you told, OTU where it is there? In which file it is there? No, you are having QZB, where OTU is there? Please understand that point.
This one.
Next step, what you are also running, next step, yes. Yeah, next step, yes.
This one.
I just tried it and
After this, yeah, you have done. Next step, go to, yeah, give the next comment. Yeah. Now we have to prepare a TSV file. Because here only you are generating future tables. Yes. This is denicing. What is the use of denicing? Removing errors. Denicing, removing errors. This also told that lecture you are visualizing.
This also has been done. This.
Future tables. Next. So here only you are giving the taxonomy. You are visualize that.
Now, sir, they can see. Now we have changed that part. Okay. See that denoising steps. What we have done. Previously, we have given the for reverse and forward read. We have given 200 in the left. Yeah, right, and 23 in the left, right? Now that part we have changes. From 200 we are keeping now 240. We are increases because previously we are getting very less number of features. So now we have changes, increases that part. After that, what we are getting now, you can see the difference. You can open your table.qzb file and this one also you can see the differences. Now the feature.
They haven't done.
They have done that part.
Yeah, they haven't changed that.
Yeah, I have done. They can see the difference. And now it takes time to run. That's why I done
You
here.
have to show that thing. You
Yeah.
have to see them.
Go to that table. Yeah, here it is. Go to interactive.
What is the
Yeah,
command
yes.
you have given?
Yes, yes.
Nothing
See
is
now.
there. Command.
Comment. Go to the comment. Yes.
Yeah. Please note that thing. See, Previously you have given 200 forward.
Now in this way,
Left side 23 you have given and 220 you have given. Right? See how they have changed. 240, 210 and 230 10. So like that, tomorrow if you are getting this kind of thing, manipulate.
this is the troubleshooting. Okay.
It's called troubleshooting.
Right?
Debugging, troubleshooting.
Now you see this thing. See now previously 30
It was 2.
5 like that. See now you
Now
see.
see.
Yes, lot of improvement is there. Right? 77,000 you have given.
Now do
Previously-
the next step.
Previously we have got only 7,000. Right? See? So that's why you should know. Yeah. That's why sir clearly told about that trimming very important point. Very important. Okay, now with your data you continue what?
Yeah.
What you are doing, just go
we
with that.
just want to
You
see
do
the
that and we will compare with this.
Yeah.
Sir,
Okay.
the next step, this
Ah.
frequency, how we are doing. Features, filtering the tables.
Which one?
वो कम दिया मैं को नहीं तो नंबर तो चेंज कर लेकिन कम दिया
One feature table we are filtering. Like, this frequency is constant or we need to.
No, you have to change it because you have a minimum number of 2. So if you give this, everything will be deleted. Okay, nothing will be left.
Check the data.
Oh, then to this thing, the feature table. No. Go down. So this value need to be changed. Okay, because if they keep this value, everything will be gone out. Okay, because this needs to be like 2, 3 or something because we have got very low count of data. Okay.
so the minimum and maximum the minimum frequency
This is minimum frequency should be
Yeah,
low awareness. Look for that frequency. Minimum, they don't need to do these things.
Yeah.
Okay. So remove these filtering steps for the time being. Okay. Don't do the filtering steps. Don't do the filtering steps.
Okay, then what they are doing?
So don't just go for the assigned action.
Oh, straight away you can go for assigned taxonomy.
That's the important step. Because we got very few numbers of features, so we are now just not using these particular filters. There is no need to use this filter. So this is a pretty good filter. One is based upon the filter feature table, total frequency based filtering. So here you can mention a minimum number of frequency you can look for. And next one is the low abundance filter. What we did yesterday with microbiome analysis, can you remember 4 or 5? That thing is here. Its minimum frequency is 10. So just simple. It's the same thing like the microbiome analysis. Okay. Now go for the assigned taxonomy.
So for that, you just go for 3.1, perform taxonomy assignment. Because we have already downloaded this classifier file. Because if classifier file is not there, you can wget and then you can give the HTTP.
So I am changing silver database.
You can download that.
I have to change
You can either download or you can have to create this thing. That's creating this thing is
Here
another
itself,
kind.
Kindle itself will recognize.
If you are giving this message, it will do that and then
Here you are not. You are giving classifier.
this is the classifier file you already have, sir.
Classifier file we have now.
Yeah, classifier. Yeah.
Yeah, we have.
Suppose I want to use silver.
You can change, sir.
How I change?
That change you first need to train the data set. So you need to download the whole 16x, they need to train and that is so. So this is another big long process. That's why I just, because then that will be too much for them to grasp. Because if they do it now, the whole thing, now they can later on which can be introduced.
But in green gene database, everything is there.
No, green gene is obsolete now. It's old.
You have to use.
Silva is good, RTP is good, NCBI is good. Like what we did, we actually NCBI, Silva, then RTP, they all have some unique sequences which is not present in others. We combined 3 of them and created our metadata.
Here you have to train all this, huh?
You have to first cut the region, like you have before region, then you have to cut the before region. If you have before region, then you have to cut the before region. Then based upon that you need to train your classifier. You look, this before region is for these organisms. This before region is for these organisms. This will be the training.
Means you have to know everything.
I mean it's like machine learning. It's like machine learning. So you are training the program. Then SK classifier that already learned that thing. So based upon that training, it can recognize your new sequences and designate its source.
For this, we need a skilled person.
No, but it's something else because you need to download the whole database. Now then you need to treat them. Okay. Now you need to take import it into the time to. Then you need to learn. So this is a long process. You don't need it. You can whoever can do this.
Try tomorrow, sir.
Yeah.
Then if it is
So
not working, then
yeah,
we can call you.
it will work. Don't worry, it will work. People are doing these things. So I am not the one person who will do these things.
For us, it's very new.
You have always existing in policy.
You can call, yeah.
No, they have already done that hybrid thing. That link if you are having
So don't worry. I mean, anything can be done. But putting it here, it will be too much for them.
Yes, sir.
whenever you are publishing paper, is it okay?
The GG, I mean, again, is now older. So you have to use some other, like Silva is there, we are testing, RTP there, even NCBI database is
Anyone available in Google search like this?
yeah, some of the classifiers are available, like Silva classifier is available, NCBI classifier. We can use for B3, B4, all these things are available. You can directly
Download and we can, instead of this, we can do that.
This one now.
Same taxonomy,
Yeah.
Now look at it. Now you have to look at it. Go down.
Go down.
Haven't performed.
Yeah. That is one.
Yeah. Now you have to create the taxonomic table
and the visualization.
and you have to visualize it.
To visualization, you can.
Can you save features?
You can see that this alpha, beta, so that they will Now, look, this is very, now they have to give the time. It's not that spoon feeding is not this thing. Now, you have to spend time and you need to learn each of these meanings. What this feature classifier, what are the different options are there. You need to learn.
Give it this one to compare view set A, view set B, and then you will visualize.
That's the most important.
Enter this command.
But I think if someone is interested, he or she can use to follow these things and use to explore them, they can do it. No problem. Because this is now this very standard. A later part, like whenever you got this table, then you can either use the micrometric or directly just take it to the art platform, use the phylosync or something. Phylosync is a that is, I can show it. Go to the bioinformatics, just type right, go to internet. and write it according to one format. Yes.
The same thing here also. The phylogenetic tree builder. Sorry, alpha diversity. See, dash dash that I dash double. After that, you remove this part. Filtered sequences slash filtered up to dash. Okay. Don't give that one, and it should be table dot QZA, not table 2. Okay. The other thing will be similar.
Then output the
Other thing, yeah.
filtered it will be.
Yeah, filtered sequence, automatically it will create one folder, okay?
Or should be
Input, up to input, right, understand, right? Then output also, the filter, that part you just remove. From down also, okay? Up to filter dash that, now, and keep it as table dot QZB.
It will be replacing the previous table dot QZ.
Yeah.
Okay. The same
तो फाइल को क्या करना आपको फाइल को
filter sequence table input table then table dot QZ dot QZ.
हाँ सब रहेगा, सिर्फ वो ही नहीं रहेगा, बाकी सब रहेगा.
Same output also table dot
Still setting will come to you, don't worry.
that there also like table dot sequence
ऊपर जाओ कमांड to sound.
No such option.
यहाँ पे आपको यहाँ पे space दी table dot QZ है ऊपर.
ये no such options.
Done, right? I
So
see.
this is
Wait 1 minute.
It doesn't exist anywhere.
Wait.
Already present.
Let me do it here. Okay. Must come. Otherwise, the Nisha make directory you keep do make directory filter dash this one. Try once. Then filter dash sequences na. You try make one directory. This one na. Make mkdir filter dash sequences. This one we are doing na.
in that
Oh, because your edified user doesn't comment. What is the edification?
So I think we have to skip that part.
Yeah, you should skip that. Don't use the edification because we don't have enough sample. Okay.
So
Please, yeah, you have to skip that part.
which steps, sir?
The edification step. Here, this step 6.1. Okay. Or you can simply make P sampling depth, it's 2, okay. its value you can make it 2. That you can do, okay. So instead of 1300 you make it 2 and run it again.
Nine. Can access
Yeah, it's done. Now open the directory. Now go to the directory.
It's done though. You have done some mistake. Give me.
Yeah, diversity matrices. How much did you
Now you open that one.
Yeah. Now open this QZ. Just look for this files. Okay, first. Yeah, only the QZ files. Then you can see the breaker is diversity. Diversity. Then you also get the alpha diversity there. No, not that one. So first try with the business or channel. No, it goes there will be no. Channel breaker is given. Okay, then it has breaker. Breaker is what? This is a emperor plot. Now go to there, select a color category. Select a color category. Yes.
Done. आपका हुआ?
Then sample ID.
हुआ सबका? Table.qz be generated.
Yes. Now it's working. But data is not good, so she got this emperor plot.
Yeah.
Just make it 2. Okay, P sampling depth that equals to 2.
Minimum.
Then minimum. Then do the alpha radifaction. Then you will get the radifaction curve. Radifaction curve the Shannon thing you can do. Okay, just see these things. Radifaction thing. Just keep. You can do this one.
By changing to 1000 rate.
That will be.
So after alpha diversity,
Yeah, you do this thing first. Okay, here you will get a directory diversity matrices result. Within that you will get everything. Okay, from where you will do the next thing.
something that will give this one result.
2. Look, she got these things. Access 1, access 2. Now go to the sample ID. Change that thing sample ID to.
The sampling that on what
Stages.
this?
Go to the stages. So now you can see one is healthy, one is infection. But as the quality is not good, so it's not different. Healthy and infection, they are merged with one. See this?
Sir, on what basis you to fix something that's minimum frequency and maximum frequency?
Which one?
Is something that?
Sampling depth. So it's mostly based upon the minimum frequency of any sample. Say 7,000, then take it to
Minimum frequency
7,000.
only have to
Below that.
In that one, I have said 600 is the minimum.
Then you can use the 600.
600.
But ideally, because there is too much anomaly in the so difference is there. Ideally, this doesn't happen. So if you get 77,000, so the lowest one should come like 33,000 or something. So you can take up to 30,000 or so.
Put table, gap, table dot.
If something happened with this data, it's not a good data. This is free data, na? That's why they are giving this free data.
हो गया वैसे?
इसे छोड़ दो अभी. Go to the next part.
Go for the that this one, yes, yes. 6.1. Generate alpha and beta diversity matrix.
2, right?
The next step, na?
Now I think they can do it.
Yes.
Yeah? You have some bit of confidence. Nick, do the Shannon index. Shannon group wise you do, and if possible do a OBD one. These two. Faith PD. So for this you have already generated this
Give
file
2, okay?
in this folder.
Depth, give 2. In place of 13,000.
Sample wise you can color it.
I think they know.
Go to the color, not like that. Don't change all the things. Go to the color first.
Yeah. Two.
White.
Only two.
Yeah. That's fine. Yeah. It's a day one stage. I think the infection one. You can go at that stage, go to the color and the stage and look at the sample wise. No, not there. No. Go the stages. Yes. Look sample ID. Click the sample ID.
The third one.
So you can see sample ID wise different color is given. So this is the emperor plot. Yesterday you have seen this thing, okay?
Yes.
In microbiome analysis itself you have seen this thing.
Yeah.
Use the QZV, not QZA. You need the QZV files. Okay. In the diversity matrix results, you will see the QZV files. Only you can see by the kind view.
Sir,
Yeah. Where you got this? Yeah. You can move it. You can just.
Drag it.
Yeah. In every direction you can move. And you can change. Select a color category. You can change sample wise, stage wise. No, go to the color and below there, yes, select the.
QZB फाइल है ना, आपको ड्रैग करके देखिए.
Yeah, it's done now.
हाँ, QZB एक्सटेंड फाइल टाइप है ना, QZB जो है वो अलाउ करिए वेरी फोकस नहीं एक सेलेक्ट एंड देन ड्रैग इट, यस. बड़ा कीजिए उसको भी, मैक्सिमाइज कीजिए. तो अभी देखो यहाँ पे select the color category. So on what basis we are going to show this plot? Barcode sequence wise. So we don't want as a barcode sequence wise, right?
And then
So we want stage
this
wise.
another step is there.
So your healthy is showing us red and infectious is showing us blue.
No,
So this is what we are doing. So you can see this part. Okay? But the quality of the data is not that great. Very small number of reads are there. Okay? So you can change the color also.
Here you can do the face PD and then SAN.
And then visibility also you want as a stage wise.
You can later do it with just changing the parameters.
Stage wise.
The whole thing.
Scale you want stage wise. Shape you want stage wise. Axis. Then you can change the background color. Okay. Background color where normally from white, right? And the label you keep black.
This is free sequence is not also good for you.
So we are having axis 1, axis 2, axis 3.
She got so somewhere there is a segregation, okay?
Yeah.
Blues and reds.
Yeah.
But they got something else. I don't know.
So this is some separate folder. QZV instead of QZV.
No, it's the same folder.
not QZB.
Same data. Breaker is, yeah.
That's fine. They can,
Yeah, it's
I
fine.
think they have done. Yeah, like that you can change these things. Size
Look,
and-
there is some difference actually.
Okay.
So you need this thing?
Right. Understand, bro?
Yes. Fit PD. Yeah, there.
See, these are because of this the barcode, na?
So this is
So you are
like
segregating
barcode
barcode wise, na? Like because of that only.
Stages.
Do always these stages.
The diversity is pretty high, but for these, infection diversity is low. That's what is expected. Look,
See, here the results. This is empirical.
Vinata, just look here. This is the faith PD.
Yes.
And for the LD, the diversity is high. And for infection, diversity
Is
is
less.
also do the Shannon one. That's the last one. Okay. So here you need that phylogenetic tree. Unless until you create the phylogenetic tree, you cannot.
Now I finally
Done. So open the other file also. So see here they have for disease. See. This is your infections. These are healthy. So these are forming somehow one cluster. These are forming some other cluster. Visualizing or you can see this one. The other one also. The other files. Because there are so many files. You just open those. Sister. See. So your thing is not same color. You have to do like this.
today you get 19.
So if you have any problem in future, please contact her. Okay.
No!
She can contact me. She is now an expert.
No, sir. Still so many things.
Jinata.
Yes.
Look, everybody is calling you. Okay.
You're comfortable, sir. Yes. This diversity matrix is open. See, all the QZB file are there, right? See, dot QZB very cookies, right? And dot QZB is record. And then dot QZB weighted unifragment period load.
Yeah, Moto is calling you.
One more.
Oh, Moto is calling you.
After in class shows, someone is giving the evenness index, someone with the giving the similarity index, like that. So you have to check one by one. Different indexes are giving different.
I just want to see the Shannon one. Shannon one, I need to see.
Not ID wise. Always do stage wise. Or not stage, whatever you are giving in metadata information. Yeah.
So one thing you can do, you can change in the metadata and upload it again, and you can again see all these things. It will change the different stages you will get.
Now open the another one. Once you have done this one, open the checkout one and see how to check the Shannon also. Shannon want to run it. Sami, working there? Already this one. Now you already have done the. See now, see you have to go to here. Okay. Now here see QZB file are there. This one is QZB file and this one is a QZB file. So 1, 2. Now it is show then just drag it. That shows what. Okay.
Why we just get it?
Just drag it.
You can also change those numbers.
One of the elements which amount of data for this you are doing.
Everyone's different.
So fine. Inada, she already also got this Shannon bar. So it's fine. Okay. So everybody will get it. Emperor Pope God you already got.
They are doing it.
If you type this one, what? Alpha.
Because na
Alpha layer.
because see आपने ये कॉपी कैसे किया था ये एक मिनट ऑलरेडी ये डायरेक्टरी मिल रहा है
Yeah, go to this select. Oh, you done. Oh, yes. Yes. Now go to the select color category here. No, there here go down. Yes. Now select stage. You can both do stage wise and sample wise. Now you can see one is healthy group, one is infection. Okay. You can move it also. Just drag it. Yes.
Sir, I was in only before QZ
Done.
if you want to see the
File.
space.
But you know here you have 146 features, so you get that huge 146 nodes. Okay, no need to see. But yeah, you can. But for phase 3D and later on, I think weighted, unweighted and weighted unit type is also there. And slashy there. Yeah. So for this you because the unified distance they will consider this value as a whole slash. And then I am leaving. All of you can do it. Okay. Okay. Thank